All right. I think it's just a minute after 9 by my clock, and I know everyone's clocks are a little bit different, but hopefully that's pretty close to what we are all on. I'm going to be giving the talk today. I know I introduced myself last week, but I know some folks also weren't able to make it. So just as a really quick introduction, I'm James Keach. I'm the extension agent for master gardeners as well as ornamental crops and landscaping over on Kauai. I worked on Big Island before I came here, working on kalo and uala, or taro and sweet potato, as well as a little bit of work with koa. I've worked in Singapore before that, doing orchids for a garden there. And yeah, my background is in plant breeding mostly. I bring some of this up because when I was doing my PhD, one of the things I did for money, just what we all have to do to get through a degree, was TA plant propagation. And I bring this up especially because normally plant propagation is a semester-long course. We go in depth in all of these different things. There's a weekly lab. And so I'm going to try, and this is a theme you'll hear from pretty much every speaker in one way or another, I'm going to try to condense a semester's worth of things into a few hours of talk. My goal is not to make you completely fluent in plant propagation from this. My goal is to introduce you to certain concepts, certain words, so that when you come across them as you're looking stuff up, as you're reading things, or someone mentions something to you, you don't say, well, what the heck is that? You actually recognize it and say, oh, okay, I sort of remember what that is, and can kind of approach it that way, and then look it up some more. When I was learning the sciences, one of the most influential professors for me, his technique was always, you can look stuff up, but you need to know what you're talking about to look it up, what the right word is, and all that. So this presentation is kind of made in that vein. Kind of a few housekeeping sort of tech notes. I have this on two screens, one of which has my presenting slides and y'all's faces. So if you see me looking to the side a lot, that's what's going on. Also, a gentle reminder to mute if you haven't already, just because we have 70 participants in this. And so, lots of background noise can kind of get in, disrupt things. And I know my other fellow coordinators will be kind of monitoring and helping mute folks who forget to do this. I think that's most of the stuff, and I'll get on with the lecture. So I put this photo at the beginning because I think we typically think of plant propagation as something that we do to plants, but plants are also perfectly capable of propagating themselves. Some of them do it by seed most of the time. Some of them do it by non-seed forms. This is what they call mother of millions, where it's forming little plantlets around the periphery of the leaf. Each of those can fall off and form a new plant. It's really cool. They make great weeds for that reason as well, but they are cultivated a lot for it. So I think, we're using our own technique. Sometimes we're improving on nature, sometimes we're just copying nature. I think it's worth keeping that in mind because we can learn a lot from how we see the plants handling things also. So the way I'm going to be going through the talk today is I'll split it up. I'll have breaks in the middle, but roughly this is how we're going to go over things. Talk about what plant propagation is and what it isn't. Talk about a few general terms that are kind of applicable to different fields of plant propagation. Take a break. I'll go into asexual propagation. Propagation generally that doesn't involve seeds is how we'll define that. And then take another break, and then we'll talk about sexual propagation, which is mostly seeds, and some of the exceptions with that. We'll have a little bit of time for questions at each break. If you want to put questions up in the chat, feel free to do that throughout this, and we'll kind of try to address them during the breaks. But as I mentioned, please keep it muted just so that we can all kind of move forward with this, because I know there's a lot to learn, and I want to make sure that all 74 of us now can, kind of learn the most from this. So we'll start out by talking about propagation. This is kind of a general, very general definition. Basically, when we're talking propagation, we're talking about multiplying a plant, boy, that's a bit of a tongue twister, normally in a way that keeps it true to type. And you'll also hear folks talking about reproduction. There's also a great short musical interlude in Grease 2 titled Reproduction. So not very informative from a scientific point of view, but if you want a fun thing that covers some of the things we're talking about today, it's a cute watch. I talked about this a little bit, but two typical forms of propagation. We'll talk about asexual from plant parts besides seeds, and then sexual from seeds. And the biggest thing to keep in mind with this is that most plants technically can be propagated both ways, but most plants also have a way that's much easier than the other. And sometimes it hasn't even been documented that the plant produces seed or that the plant can be propagated asexually. So that's part of the learning process too. And I'll go over that in kind of this next section, what you're looking for when you're trying to propagate. So asexual propagation, I'll go into a little bit of detail about things on cuttings, different types of cuttings from different parts of the plant, air layering, something you might have heard tossed about as a kind of technique, tissue culture, also known as cloning. And I'll get into a little bit more detail about that just because it's something you're less likely to encounter. So I want you to have a little bit more background if you do hear about it. And then grafting. And grafting, in the before times, we had a whole training on this. It's a little bit harder to do. So I think this is probably going to be an advanced training outside the course, which is a good reason to kind of keep involved so you get to hear about this further on. But I'll talk about some of the things you can do with grafting and the basics of it. Sexual propagation, the later part of the talk, almost always is from seeds or from spores if you're talking about things like ferns. I'll talk a little bit about how we test for germination, things like seed dormancy that comes up a lot. Why is my seed not growing, essentially? How to save seed, and we'll be doing an advanced training on this, I think probably in April, that I'll get more details out to y'all about. Hybrids, what they are and what they aren't, seed treatment, how this comes up, and then how to store seeds safely. And, again, very kind of general talk about it. So first thing I wanted to know, I have to actually open the chat box now, is what sort of things do y'all want to propagate? Let me know, and sky's the limit. I can try to kind of let you know some things if I see them popping up. I haven't seen anyone put anything in yet. But if there's a specific plant, lilikoi, excellent, thank you. Lilikoi is interesting. It's a little bit challenging to propagate from cuttings. Normally needs some heat below the cuttings, but then will germinate. You can do it from seed, sometimes germinate faster than other. Great, now they're pouring in. Succulents do really well from leaf cuttings. Ohia, yep, you can do air layer with it. The only thing right now is air layering does involve wounding, and so rapid ohia death can actually get in that way. So I've seen a lot of folks encouraging people to do it from seed. Coffee from seed, from grafting. Noni from seed, also from grafting. Saving avocado trees. Grafting more common just because you'll get something more reliable. Yeah, just looking at a whole bunch of stuff here. I'm not going to go into each of these, but seeing a wide range of different things that we can propagate in different ways. Papaya, yep, one that I actually have an example from, and hibiscus. Hibiscus is one I always find more challenging to propagate from cuttings than from seed, but it doesn't come true. But grafting hibiscus is about the easiest thing you can graft, and it's really forgiving with it. So that's a good way, pick up a cheap plant at somewhere and use it as a rootstock and then graft something else on it and teach yourself kind of grafting that way. Excellent. Yeah, I love seeing these kind of examples. All right. Okay, so choosing a plant. I encourage you to do kind of some background thinking when you want to propagate a plant. You want to choose the best plant if you have a few of them, and you want to make sure that it's healthy enough you can support it in the best way to make sure that it survives propagation. It's not always the best thing if you go to propagate a plant and then kill off the original plant, because then you just have one more plant. And sometimes, there's reasons to do that. But in most cases, you want to take a look and kind of think about a few things. So one thing, plant health. Is this plant healthy? Down here, this picture, we have some pretty unhealthy tomatoes and some pretty healthy tomatoes. I would be trying to propagate from those healthy tomatoes. On the other hand, maybe you want that, you know, really good variety, but really unhealthy. Kind of up to you. Another thing, size. Can it support losing branches? If we took a cutting of this plant over here on the right, that plant doesn't have a lot to lose. It might survive it, but you might want to kind of nurture it a little bit more. But you do see kind of a new shoot coming up. So maybe, you know, just kind of thinking about these things though. Is there a disease that it has, again, with these tomatoes that you might be propagating in addition to the plant itself? Are you cleaning your tools so that if it does have a disease, especially a virus that you might not be able to see, you're not accidentally spreading that to the next few plants that you work with? How do I propagate it? I'll talk about this a little bit, but the thing I'm going to say, you know, since you saw with the chat, just so many different things people want to propagate, we can't go into each of them. Do some background reading. A lot of plants, you know, you'll find that there's really good resources out there about how other folks have propagated them. And sometimes, like I mentioned at the beginning, you can propagate 2 different ways and just get 2 very different results. A photo here I found on the right with tomatoes, where if you take it from cuttings, you're going to get fruiting, flowering much earlier than if you're growing from seed. On the other hand, tomatoes here get quite a few diseases. And again, you know, if you're propagating them from cuttings, you might be propagating those diseases too. If you can't find literature looking for certain pertinent characteristics, I'll talk about different types of woodiness, things like that. Being honest with yourself about your setup for propagation. Do you have enough that you can actually, you know, do the right form of propagation for this? Or do you have to kind of like figure out stuff? And there's all sorts of different ways to propagate. And then I mentioned it before, and this will come up several times just because it's something I think we tend to forget about. Is it at risk of having a disease that might, you know, not be visible in a healthy plant, but in a cutting where you're cutting it off from its root system, you're going to have that disease kind of flare up or spread to your other plants, heaven forbid. Another thing that comes up, I feel obligated to put this in, especially as a university employee, do I have permission to propagate it? Some plants are patented. I'm not going to get into the ethics of plant patenting here. That's several talks worth of things. But be aware of that, especially if you're going to be selling plants commercially. Don't get arrested. When I was looking for pictures of people stealing plants, there were a depressingly large number of things like this with folks getting caught on video cameras. This botanical garden's always struggling with this. This was a photo a friend took at the San Francisco, the Striving Arboretum, and had to embed all their plants with GPS tracking chips. I don't know how true it is, but you know, they were obviously upset enough to have to put up a sign saying this. Also, this is a term I only learned in the last year or so among some of the online forums of propagation. Proplifting, kind of a play on shoplifting, is folks who will go and sneak leaves, pieces of plants from a nursery or from, you know, a big box store and try to propagate it. There's a lot of controversy about that. As a university employee, I'm firmly on the side of the law. Don't do this. But I think the biggest take home with any of this is please don't get arrested. The one thing I didn't get a picture of for this, but is also worth keeping in mind, especially here where we have so many endangered native plants, is that you'll see a red tag on a lot of them. And that red tag indicates that they've been propagated without damaging the original population, you know, that they were propagated from a nursery. So it's worth looking for that. And also, if you're going to be selling or giving away plants of that, seeing if you can get more of those tags to show this has been kind of ethically done. And these aren't being ripped off a hillside somewhere, messing up the conservation efforts. I'll try to get another photo of that and maybe send it around later, but I just couldn't find my picture. I know I have it somewhere. Is it worth it? You know, yeah, for sure, propagation of some plants is easy and satisfying, and that's totally valid too. But also, you know, this picture of red geraniums over here, you know, I enjoy propagating. Geraniums are easy. They're really, you know, satisfying, but they are in most cases very cheap and easy to find. You know, is it worth all the effort to do that? Yeah, satisfaction, maybe it is. On the other hand, if you go to Akatsuka Orchids over on Big Island, this is a lady slipper valued at $20,000. If you have a plant like that and you can propagate it and then maybe sell a cutting of it, yeah, I'd say that's well worth, you know, propagating. Also lower right here, the blue poppy from Tibet, Mechanopsis, is really beautiful, hard to find. If you have that, yeah, you can probably call in some good favors, maybe even make a little money if that's your thing off of propagating this one. But also thinking again, coming back to plant health, is this something where you have the healthiest plant, or would it be easier for you to buy another healthy plant and start with that? I spend all this time talking about propagating from plants that are healthy, and then I put up this slide about you can actually save plants if they're like one of a kind or something that you really want to hold on to, if they are injured, diseased, etc. And you can do this by various forms of propagation. Cuttings, you can see on the far right here, this poor pine tree has been very much abused, excuse me, but is starting to put out roots. And you could probably cut that off and stick that in the ground. It would probably be happier than its current situation. I've seen people have a papaya variety that they like that's maybe different from what other folks are growing, do air layering, tomatoes. Down here on the lower left, you'll see a tree that's been girdled. This happens a lot with beavers, sometimes with rats. A little bit harder to do, but there's actually a grafting technique where you can graft from the bottom to the top and kind of reconnect some of the nutrient flow like that. I tried to find a good picture of it. I think in the Grafton later it comes up, but it's a really cool technique. It's not practiced that often. But there's all sorts of things you can do. If you have a special plant that has some, meaning to you, you can use propagation as a way to save it. I'll get into growth regulators. I'm going to go into specifics on this for a few slides, but I wanted to kind of go into kind of a general talk about this first. You'll hear plant growth regulators abbreviated to PGRs, especially in the industry. A lot of nurseries, they'll say, oh, what PGRs did you use? A lot of us, I think, we kind of hear it as hormones. Technically, the difference between these is that a PGR, plant growth regulator, can be either naturally derived, like an isolated from a plant, or it can be a synthetic chemical that's created some other way. A hormone is always one that's produced by the plant, and so it's found in plants. Some folks you'll see kind of saying, oh, this has a natural advantage. There also kind of is a flip side to this, though, is that if the plant produces it, the plant often has a way to metabolize it and get rid of it. And so because of that, the hormones tend to not last as long as synthetic plant growth regulators, where the plant just keeps reacting to it. I'll go into detail about some more of these, but some that you might be more familiar of is auxins. These are rooting hormones. A lot of times if you see like general rooting powder or rooting dip, we'll go into that. Cytokinins, these are things that induce shoots. Orchid world, this is pretty popular for things called keiki grow. It was the first time I'd heard the word keiki was on the mainland talking to orchid people. But there's a whole bunch of other ones, and I won't go into all of them, but I'll go into the ones that you might be encountering in propagation stuff. The other thing is this is very much a dosage dependent thing. A lot of these can cause the plants to grow themselves to death. Actually, some of these, and I think I'll get into this on Gibberellins, were discovered because they were causing death in plants. And in the case of Gibberellins, it was from a naturally occurring bacteria. And also, speaking of naturally occurring, plants create these, but bacteria do also. And so this kind of lump of growth that you see on this tree is what's called a witch's broom, and this is created from bacteria called Agrobacterium tumefaciens. It's used in genetic engineering. They're taking advantage of kind of its natural genetic engineering, where it'll inject the host plant with this string of DNA that causes it to produce hormones, but then it also causes it to produce some amino acids that the bacteria needs to grow. And so this was kind of something that was observed and then was later used by scientists. Okay, so big disclaimer as I go into this, and kind of disclaimer to my disclaimer, I feel I should say I work in organics quite a bit. I worked in organics. I did my master's on organic options in wheat. I did the first half of my PhD in organic vegetables. I want to kind of clear up a few things when we talk about things like chemicals, what that means and what that doesn't mean. Partially because I don't want y'all to get in trouble, but also because I want you to kind of walk into it with open eyes of what you're doing. First of all, always read the label if you're applying anything. hormones, PGRs, fertilizers, pesticides, herbicides. For one thing, it's illegal to use them without following it. And this is for your safety, but this is also for the environment's safety. And this includes organic options. I wanted to specifically bring that up because over here on the right, we have something called the environmental impact quotient. And this was derived from a bunch of researchers who wanted to see how different chemicals affect the environment. And they look at the active ingredient in a bunch of different applications. I think, myself included, when we hear organic, we tend to kind of think, oh, this is better for the environment, this is better all around. That is true in some cases, that is not true across the board. Bordeaux Mixture, this is one that was used in grapes a lot, still is used in grapes for organic production, has one of the highest impacts on the environment of any fungicide. And so, I think when I looked at this, it was a list of something like 30 different chemicals. Bordeaux was number 2. So there was only one other thing that affected the environment more. Again, not trying to bash organics, but saying, be aware as you're looking at the different chemicals here, how does this kind of affect the environment without just seeing that word organic and thinking, okay, this is safe. And again, for your own safety as well. If you want to look up environmental impact quotient, I have the link down here at the bottom. I also have it at the end of the presentation. And when you send out the slides, it should be clickable. Another thing to keep in mind, some plants are sensitive to some chemicals or not. When I was working on impatiens during my PhD, there were very specific things in labels about if you apply oils to them, you will cause them to lose their leaves. And I, one time, have to admit that I did not pay close attention to that, and sure enough, ended up with a bunch of leafless impatiens. So be aware of that as you're kind of looking around. Not every plant reacts the same way. Also, some chemicals can't be mixed together. They'll interact with each other. Sometimes it makes them non-effective. Sometimes it makes them toxic. So just kind of be aware what you're working with. And as I mentioned before, dosage. Some of these chemicals, if you apply enough of them, they will, cause a plant to kind of grow in a way that kills itself. Same sort of principle as water. I know I talked about that in the intro last week. But just kind of, trying to drive home, do your research, do your background reading when you're looking into different chemicals, organic, indigenous, or conventional. Please, please, please. All right, all of that aside then, we'll get into different types of plant growth regulators. So the first one is what are called auxins, kind of like if you're talking about a bunch of cattle, that pronunciation. Common ones you'll find are 2,4-D. This is used a lot in commercial applications. It's used less as a rooting hormone and more for this fruit here in the middle and other fruits like this to sometimes cause smaller seeds and larger fruits. This is a naranja. It's a South American fruit, pretty good for making different sorts of drinks. But it can be used as a rooting hormone. NAA, naphthalene acetic acid, is pretty common as well. And then the other one you'll see a lot of is IBA or IAA. And these are very similar compounds. I believe they're both hormones, whereas the top 3 are synthetic. A lot of these are used as rooting hormone preparations. Some plants really like them. Some plants really do not. I'm blanking on the plant right now, but again, there was a plant I worked with before where if we used rooting hormone, it would always kill the plant and it would root fine without it. Other plants, I have tried and tried and tried to root them without rooting hormone, had 0 results. Use rooting hormone and you get better results. So just kind of being willing to play around with that. And you can see down here at the bottom some tests also. Some plants will root without rooting hormone, but will root better with the rooting hormone there. Another thing to keep in mind, a lot of commercial rooting hormones have other things mixed in. Antifungals sometimes, because again, you're wounding the plant. And I want you to take from this bottom left, this mint plant here, I want you to take this away as a thing not to do. If you are dipping that plant into your container of rooting hormone, you are introducing any bacteria, fungi, anything else that's on that plant into your supply of the chemical. And so then any plant you propagate from then on potentially is getting those as well. Best thing you can do with rooting hormone powders, liquids, anything like that is pour it out into another little container and then use that to kind of use your plant and then toss if there's extra. That way you're not contaminating your supply. Cytokinins, I mentioned cakey grow. I was trying kind of blanking on other commercial applications for these. That's the one that comes to mind. If you've grown Phalaenopsis orchids, like in the upper right here, you'll see this kind of keiki coming off, and this was popularized by the Hawaiian orchid industry, which is why it has a Hawaiian name. Sometimes this will happen instead of a flower. And in this case, this is happening because of hormone concentrations. An enterprising grower realized this and actually made a paste that you can put on your Phalaenopsis stalks on some of the lower parts, like right around here, where there's these kind of, they call them scales covering up a little bud. You put it on there and it'll produce a new plant. It's also used in other types of orchids a lot, and it sometimes also can be used to kind of prevent leaves from senescing. I've seen some work in this with florists and other leaf installations using foliage, and you can see the difference between treatment and not. Excuse me. Naturally occurring ones are kinetin and zeatin. Kinetin I see once in a while. Zeatin is fiendishly expensive, pretty rare. I've used it a little bit, but not worth it cost-wise most of the time. Benzyl aminopurine, or BAP, is really common in cytokinin preparations. I believe it's synthetic, but don't quote me on that. Actually, I'm not 100% sure about that. I know thiodiaziron is synthetic, and so it is not degraded. And I've used that quite a bit. That's one where you get like this tiny vial and it's enough to last you for 30 years. So, again, these are ones you might encounter if you're looking at cytokinins. Ethylene, this is one you might be a little bit familiar of from a grocery store. This is normally a gas. It's also can occasionally be sold as a liquid that converts into a gas, but even in a lot of plant research, we'll see it being used as a gas like that. This is what they use commercially to ripen bananas. So they ship bananas green when they're really kind of hardy and easy to, ship without getting bruised. And then they, upon arrival, treat them with ethylene, and so they ripen there. It's also a result of combustion. I've learned this the hard way, unfortunately. So this whole talk is going to be a lot of don't do what I did. So when I was in Singapore, we had someone who was pressure washing the floors of our outdoor planting area for our orchids. We were preparing for a show. We didn't realize the pressure washer was gas powered. And so we went and did all of this. And at the end of the pressure washing, we were noticing, a few plants looked a little like off. Next day we came in, found all of these flowers that had fallen off, all of these seed pods that had opened up. And needless to say, it was a real scramble to try to figure out how to get enough plants for the show. One thing I think this is really noticeable is orchids. Typically in orchids, you'll see kind of one flower sort of dry up and fade off, and then it'll happen a few times. This is one I saw in a nursery where it had obviously been exposed to ethylene, where they all kind of droop at once like that. That's classic sort of ethylene contamination. Because it can cause ripening, they do sell occasionally things like this. I've seen other ones shaped like little apples where they absorb ethylene and prevent it from accumulating so that you can keep things kind of in the ripeness stage you want. Oh, and this bottom right is a reminder to myself to tell you it causes defoliation as well as flowers falling off. I had a professor during my undergrad who was using it for an experiment. And as I said, it's a gas, had not fully closed the tank at the end of the day, came in the next morning and everyone's houseplants in the building had dropped all their leaves. He was not very popular after that for quite a while. So, just being aware of that. But once you know about ethylene, you can also control it by things like airflow, by things like these absorbers, and just be aware that that's kind of what's going on. Gibberellins, you'll hear this abbreviated or called GA a lot. There are different forms, GA3, GA7, slight differences in how they affect plants, but most of the time, you're not going to have to worry about that. The main thing these are known for is cell elongation and growth. On the right here, you'll see what we think of as normal kind of cabbages, not really a head type, it's sort of a flat type. This is a cabbage without GA, this is a cabbage treated with GA. And you can see what a huge difference that makes in terms of plant height. This is from cell elongation, and the reason they're called gibberellins is because it was actually discovered from a bacteria in rice, and it caused these plants to grow super tall and then fall over. And I know almost no Japanese, but it apparently is taken from the Japanese word for foolish plant, which I think is something like Gibberella. And because it was causing the plants to fall over like they were drunk. So the bacteria that was caused that was also named Gibberella, and now we have different forms of it that have been extracted. It does also play a role in seed germination. I had a friend for a while that his hobby was buying old packets of seed, maybe varieties that were hard to find, and then trying to revive them by soaking them in a dilute gibberellin solution. So if you have some seeds that maybe are kind of past, you haven't been storing them in proper conditions, like we'll talk about later, this is a way you can sometimes revive them. Not 100% effective, but kind of something to kind of keep in mind. All right, done with all of the plant growth regulators for now. Environments for propagation. The biggest thing that you can do to help or harm yourself for propagating a plant is control moisture. Most plants, they need a lot of moisture support because you've cut them off from their roots if you're taking cuttings, or because they're setting themselves up, they're using that stored energy in the seed. On the other hand, you don't want so much moisture that you're encouraging fungal growth or bacterial growth, getting algae across your soil surface and choking it out. And so because of that, air exchange ends up being really important with the moisture. You can do this in a really kind of fancy way, with a irrigation setup, or you can do this with covering with a plastic bag. And I see this with home growers, but I've also been in commercial nurseries where they do this and just kind of clean out and reuse the bags afterwards. Depends again on what your setup is like, what scale you want to do it as. Fans are a really great way to improve air exchange. Here we tend to have some pretty nice breezes, but if you're in an enclosed space or if your yard maybe doesn't get some, thinking about that, also maybe thinning some of the plants around to get a little bit more air exchange can be something that helps also prevent diseases for plants. Propagation setups, it can be as low tech or as high tech as you want. You can have a glass of water and stick cuttings in, get the roots going, or we'll see if this video plays. Every year it's kind of a gamble. This is a high tech propagation setup. All right. So this is propagating Christmas cactus. You see they have kind of this thing of the different pieces of the plant that have been chopped up. I didn't have the video of that. They go down on this conveyor belt. And then it has a robotic arm that's using air to suction them up, line them up, and lines them up in the right kind of direction too. So you can see them pulling it over like that. It's moving over to the next one. So then after they're lined up in the right way, it'll go and pick them up. And you see a few make it through and then will kind of get recycled off, a few fall off on the side. But when you think about Christmas cacti that you're buying in a store or something, and you wonder, how do they get so many of these? This is how it happens, especially if labor is something that's cost prohibitive in the area that you're at. Okay, yeah, so moving over then, the next step with it is they take those leaves that are all nicely lined up, and then they move them into the flats. So it goes and again uses air suction and pulls them over and sticks them in. And they're all oriented in the same row, same depth in the planter. And so then they can do that, and then a conveyor belt takes that tray off and goes and moves it into the planting area. I am not suggesting you go out and buy this, but my point with all of this is you can go as low tech or as high tech as you want. And there's some really cool options out there. Or you could look into something like this and see if there's something you want to adapt from this for your home or professional setup. All right, I think that's enough of that. Okay. Sorry, I'm going to take a quick drink. Soil and media. I won't go tons into this because we'll have a talk, I believe, next week, in fact, from Jonathan Dienick, who is an expert in this. One thing to just keep in mind is different plants like different soil and media combinations. These are some common things you'll see on the right, included in different potting mixes. A lot of these, I think of, different mixes for different types of orchids. I've used these. Organic or inorganic, this is referring to organic in a chemical sense, not a regulatory sense. So is it something that's carbon-based that can break down, or is it something that's like sand or perlite, vermiculite, charcoal, where it's not going to break down? One thing to keep in mind with this is sometimes that can affect shipping. There was a huge bill, gosh, 15 years ago, I guess, at this point, asking if orchids could be imported in pots as long as they had inorganic potting mix. And it passed. A lot of controversy because that caused some huge problems for the orchid industry here. But the point was, it's not going to break down and be harboring bacteria, fungi, things like that, because it's inorganic. It does change base fertility. The nice things about bark, peat moss, other types of soil breaking down is that they provide nutrients as they break down. So if you have something that's inorganic, you have to fertilize a little more or provide some other nutrient source. And again, as I said, Jonathan Dienick will tell you way more than I ever could about this. Fertility, I mentioned, you might have to fertilize, especially if you're using non-organic or inorganics. So plants, again, you're cutting them off sometimes. If you're taking cuttings, they might need some source of these nutrients since they're no longer able to pull them up immediately. You'll hear NPK tossed around a lot. NPK stands for nitrogen, phosphorus, and potassium. Generally, nitrogen is the one that I tend to think of the most. This is what produces a lot of the green growing parts of the plant. Phosphorus, folks talk about generally flowering and rooting. There's some other things as well. Depending on where you live, phosphorus can be really limited here. When I was over in the Hamakua coast, we almost always had to add phosphorus fertilizer. But with any fertilizer, you don't want to add too much, or otherwise you get runoff, and that causes all sorts of problems with the marine life. The big thing with this is just kind of figuring out what the right amount is to get the plant to survive. This is a photo I found online that someone had tried fertilized versus unfertilized basil cuttings, and you can see such a huge difference. It doesn't have to be tons of it, but just enough to kind of give it a little bit of an edge. And again, kind of getting into that, what to keep an eye out for, you'll start to see color differences if your nutrients are off. This kind of yellowing is really common. Sometimes it'll be kind of this localized green like you see around the veins here. We'll sometimes hear that called vein chlorosis, vein clearing, other things like that. Again, they don't have roots, so it can be harder to draw these things up. Foliar fertilizers, some work better than others. Some things plants can take up, some plants take them up better than others, but just kind of being aware of that as you're growing. And in extreme cases, you end up with things like this, where it's dead. Hopefully not going to happen to you all. Diseases, I've mentioned a few times here, especially if you're doing asexual propagation, you're typically wounding the plant, and wounding is a way for a plant disease to get in. It's also a way for a virus to be transmitted. Huge, huge problem in the orchid industry. Most of the viruses are transmitted by shears or cutting, but there are also viruses that can be transmitted from touch and pretty easily. So just being aware of that, washing your hands between different plants. Smokers, I've never actually seen the data for this validated, but there is a long-held belief among folks who grow nightshades, like tomatoes, potatoes, peppers, that if you are a smoker, you can transmit viruses from smoke or from the smoke residue on your hands to nightshade plants, since tobacco is a nightshade. Again, not sure if it's true or not, but I have known growers and breeders who will not let smokers into their greenhouses because of that. So take that for what it is. I mentioned this with the water. Too much water, you'll get algae growth. Algae will actually suffocate the plant because it won't allow that air exchange with the soil. So if you see down here in the lower left, that means that you're going to have problems with getting the more of the air exchange to the roots. And especially if you have something that's nitrogen fixing, where it's getting that kind of nitrogen from the air, thinking about that. Use fresh media is one way to take care of this. Don't reuse media that you used for other plants. I have done this, but you also have to be willing to kind of live with the consequences when you do it. Clean your tools. Really, really strongly recommend this, especially when switching between different plants or different types of plants. And just keep an eye on it. Sometimes you can control this if you catch it early enough. Okay, And kind of my biggest takeaway from this section, and something I think to keep in mind in general with life, is failure is okay. You will inevitably fail. I failed. Every plant grower I know has a huge graveyard of plants. It's how we learn, and we try our best, and sometimes it's devastating, but sometimes it's also a really good learning experience. Some plants are just difficult to grow, difficult to propagate, and kind of accept that, and feel good if you can do it. If not, kind of, don't like lose sleep over it. Learn some more, I think, is the biggest thing you can do from this. Okay, it didn't work. Well, putting on my thinking and observation cap, what is going on? Why did this possibly not work? And how can I change it the next time? Being realistic, about what's available in your conditions, or dreaming big, but then, kind of thinking about how much you'll have to modify things to accommodate that. Asking around, you have other master gardeners to talk to now. You have us, the extension agents. We have whole connections and industry folks. So let us know, and we can't always answer it, but any, scientist, I think, worth their salt is willing to say, I don't know, but I can get back to you with that. And yeah, so I think with that, that's where I'll end things for this first section. Yeah, 45 minutes in, I think, is a decent time for a break. Before we go to break, any questions? I see the chat has been filling up, so I will expand this and try to answer some. Try to go up so I can answer from the top. Do you need rooting hormone for calo? You do not normally. It will root pretty well if you cut it off at the right stage for huli. I think it's typically about a foot and maybe, switch units, about a centimeter of the top of the corm, but it'll produce plenty of roots from that area. Cleaning tools, yeah, great question. Different folks swear by different things. Alcohol, now that we all have all this hand sanitizer sitting around in COVID life, that sort of thing works really well. Can get a little costly. A dilute bleach solution works pretty well. I know folks who will use alcohol and then kind of don't want to leave it soaking for a while because they want to move on. So they'll take alcohol and then very carefully light it on fire. That works really well too. You can also just pass it through a flame, through the blue part of a flame, but make sure you've cleaned off the debris. Otherwise, you're just cleaning the outside of the debris and not the tool underneath. Let's see. Refurbish old media by baking it. I don't have hard data on this. I guess I have seen folks pasteurize it, so steaming it, so baking it essentially with some water in it. I used to autoclave media that I wanted to reuse. This ended badly for me because you end up killing everything in the soil. And soil typically has a mixture of good and potentially bad stuff in it. So I would say, yeah, you can bake it. It also does sometimes change nutrient availability. It tends to concentrate salts. So just be aware of that too. I know clay pots, I worked at a place where they reused them and would autoclave them. What it essentially did was create a nice salt crust on the inside and outside of the pot so that all the roots that came in touch with that died. So, I would say maybe do that, but maybe spread it with something new as well. Cleaning tools, I think I mentioned alcohol. I started to mention bleach is another thing. A lot of folks will do 10% bleach for 10 minutes. So, 1 part bleach, 9 parts water, and soaking something like that. Again, a little bit more waiting time. And again, please get your debris off first, otherwise you're just cleaning the debris. Clean soil for an infected plant. You can, solarizing works really well. Solarizing, for folks who aren't aware of it, is when you have soil that's left out with clear plastic or sometimes black plastic over top. Clear's a little better because you get the light penetration. It heats it up and essentially steams it or pasteurizes it, like I was talking about before. Some things are killed better that way than others. I think kind of thinking like crop rotation might be a good way. If you are going to reuse it, for one thing, being aware that you might have problems, but also then maybe don't reuse it for a related plant. Use it for something different, just so there's less of a chance. But again, be aware you might still have some problems crop up. Clean dirt off, yep. Alcohol, torch is easy and good to use. Oil, sharpen. I worked for a professor who every day after we would go and plant things, would go into the lab and use a knife grinder to sharpen the edge of his shovel. And I have to say, I'm not that good about sharpening my tools, but it was the easiest field work I did with him because that shovel was so razor sharp, you could dig through and divide plants really, really wonderfully. The maki from cuttings, I believe so. There is, I know I mentioned CTAR publications in the talk last week. There is a CTAR publication on that. I am 90% sure. Look that up, and if you can't, I'll link to some things at the end of this presentation on how to propagate a bunch of native Hawaiian plants. There's a few databases. Composted, yeah, composting will kill some things, especially if you can heat it up, if you have a kind of enclosed space, or if you have kind of the composting magic alchemy of getting it hot enough that you're going to steam and kill some of the stuff. If not, sometimes works, sometimes doesn't. Again, kind of use with caution. Yeah, great about the air layering and seeds from Amaki. Cinnamon, I haven't actually heard for propagation. Or a ton of orchid growers I talk to use it for fungal prevention. Not saying that there is data backing that up, but there's a lot of orchid growers who do it. Aloe I haven't used. I know folks have tried to use willows and willow water. I've seen conflicting results, so I can't suggest one thing or another. Yeah, bleach is corrosive, so don't leave things sitting in it too long. Wash it off when you're done, especially because you don't want to be then exposing your poor plant to bleach since you're already wounding it. Yeah, very good reminder. Problems with Plumeria, that is Plumeria rust. And CTAR, I know for a fact, has a whole thing on that. And if you look in the CTAR publications thing that I linked to last week, there's a thing about how to control that. Yeah, CTAR rust, great. Oh, and Kalani links to it. Thank you. Perfect.
Hey, James, maybe we should focus on propagation questions so that we will have time for these questions in our pest and disease sections, because we're getting some that aren't propagation related.
Very good point. Yeah, I'll leave the powdery mildew and rust things. Keep that in mind for the diagnosticians. Make a note in your notebook about that and for the diseases. All right, I am ready for a break. I hope y'all are too. Let's see, it's 9:49. Say we come back close to 10. Let's say 10. I will see you all at 10. Other propagation questions, leave them in the chat.
Thanks, James. Awesome.
we've got about 1 minute left, I think, before we're going to reconvene. So just as a call people to grab your tea, get back towards your computer, and put any propagation questions you might have in the chat. All right, I think it's 10 on my clock. At the chat, looks like the Plumeria question got answered. Thank you for that, Megan. Propagating Roses from a Bouquet. I have had limited success with this, to be honest. I've used rooting hormones and had decent rooting on some varieties more than others. If you look up rooting roses, you will inevitably come across a technique that involves sticking them in a potato. I have not seen any documentation that this actually works, but it's really popular. So a lot of plants, if they're kind of recalcitrant to root, having something below them that's a little warm helps a lot with that. Roses also, my experience, I can tell you all sorts of things not to do with rooting roses. Fungi tend to be a big problem with them. I've seen that being a really big issue. I would say air movement is kind of something to keep in mind with them as well. Dragon fruit. Dragon fruit, I've done from seed, it takes a while, done from cuttings. Trying to think of how we did it last time. I think we just took sections. I think we used rooting hormone. Rudy, we have a master gardener here, Rudy Sina, who does this all the time. Rudy, if you're here, feel free to chime in. Sealing wax palm seeds. I think, I'm not 100% sure, quite a few palm seeds need some sort of treatment. Sometimes it's weighting, sometimes it's scarification. I think sealing wax, I haven't propagated sealing wax explicitly. I will link to a few databases at the end. Again, with all of these, if I'm not able to give you an answer right here and now, I suggest looking in CTAR publications because some of them have been answered there. Several types of citrus onto a single rootstock, totally doable. The big thing is kind of making sure you have enough growth going in different directions. Otherwise, you end up with one dominant thing, and then everything is kind of going off in different directions. Koa nail clippers are the best thing. Trimming from the opposite, you'll see a little scar mark on it. Don't trim that side or you'll cut off the root. I've had that happen. Trim the opposite side and just like cut a little bit to break through the seed coat. Cilantro, I've had pretty good success. Fungus tends to hit it pretty heavy though. Overplant, I think, is the kind of name of the game there.
I would say also cilantro will germinate best in cooler weather. Struggle in the heat of the summer.
Really good point. For those who want something warmer, culantro, as it's sometimes called, or what's the other name for it?
Recao.
Yeah, I was gonna say Pakchi Tarang is like the Thai name for it. It's Eryngium fetidum, I think. Yeah. Doesn't bolt as easily, a little bit thicker leaves, but is nice because it grows in really warm conditions. All right. With that, oh gosh, we've answered questions for a few minutes now. Okay, I will move on as soon as I get this back. Yes, to asexual propagation. Asexual propagation, there's a few different things that go on. One thing, sometimes you're working with an existing shoot that you can kind of see, sort of like over here on the right. And even before that's a shoot, you'll notice kind of a little nub sticking out there. But a lot of times with asexual propagation, you're working with what are called adventitious shoots sometimes, or roots. And especially, I think roots is one you'll come across. And this is, at its most basic, a shoot or a root forming where it normally wouldn't. So for instance, if you saw a leaf, you would not expect little shoots to be forming off the edges of that. That's just not something we normally think of a plant doing. So that would be an adventitious shoot. Likewise, roots kind of coming off the side of a leaf, not something we typically think of. Those tend to be adventitious roots. And a lot of asexual propagation comes down, especially to adventitious root formation. Another term you'll hear tossed around a lot is callus. This is wound tissue from where you cut it. This is a photo, actually, I have of a plant I have up in our screenhouse right now, hibiscus. This is what's happening is you're getting a bunch of undifferentiated cells, think sort of like stem cells, that come out, and then they can form different things. Theoretically, they can form shoots. Most of the time, we're trying to turn them into roots. I'll talk a little bit more about this when we get to tissue culture. I've mentioned wounding quite a few times. I will mention it probably a few more times as well. Big things, thinking about moisture, said moisture is key to propagation. If you are wounding, that's a big wet area that can dry out and suck the moisture out. So you'll see people doing things like putting a wax plug on top or covering it with some sort of paint or tar. Efficacy varies, but the big thing, the take home from it is you want to control the drying out and moisture for it. But also you don't want it to be too wet or you're getting diseases. So kind of, that fine balance sometimes. Not applicable to every area, knock on wood, but especially for those of you on Big Island, and I think Maui now has little fire ants as well. We have one population here on Kauai, and I hope to keep it that way. One thing, if you are doing any form of propagation, or if you are bringing in new plants at all, is to test them before you introduce them to your other plants and potentially spread your little fire ants around. I knew one researcher, who I will not name, who would actually bring a jar of peanut butter and sticks to garden centers and put them in the plant, put the plant in their cart, walk around, for about an hour to see if little fire ants came out or not, and then pull the stick. There are much better things online about how to do testing, so I recommend you look those up. This was something that Arnold Hara had come up with for how to get rid of fire ants that might be on cuttings. This does not work for media, and I bring this up specifically because this has been shared falsely. Especially when I was on Big Island, we heard this a lot, that you could soak a whole potted plant and it would kill all the little fire ants. The reason it doesn't work is because of things like surface tension, which you can see is how these ants are kind of sticking together. So if you have a potted plant, there will be air bubbles in the media. I talked about air exchange before. The little fire ants will inevitably go into those and hide out. So you can use a soap solution if you are working with cuttings. You cannot use it if you're working with a potted plant. So please, please, please test. It's the easiest way to prevent getting little fire ants. And then you can use the soap solution soap to get rid of them if you do. I use this. I brought over 8-10 suitcases worth of orchids when I moved from Big Island to Kauai. And every time I had to unpot all the orchids, take all the bark off the roots, and soak them in a soap solution and do this. It worked every single time, except the one time that I did not follow my own advice and didn't pull out enough of the media. There was a pot that had a bunch of media still stuck in the center. And sure enough, that was the time that I took it to HDOA, Hawaii Department of Ag, and they found little fire ants. And I had to go and retreat and cut off all the roots and reschedule. So learn from my mistakes. Please, please, please do not spread little fire ants for your own sake, as well as for everyone else's. Another thing that's going to come up when you're looking into asexual reproduction is variegation. Variegation is a term you'll probably hear quite a bit. this is when you're getting this kind of nice, depending on who you ask, coloration difference. Typically, it's kind of a whitish or light green with a darker green. And what's happening here is you have what's called chimeric tissue, where you have two genetically different tissues in the same plant. In this case, a lot of times what happens is plants have 3 cell layers, and they'll have one layer that's one type of tissue, normally has the normal green chloroplasts, another layer that has defective chloroplasts. I know Russell talked a little bit about chloroplasts last week, and so they can't form that kind of green color, and so it creates this really nice dappling. The problem comes up when we're propagating. Sometimes we get things that are coming from one layer, sometimes we are getting things from another, and also for seed propagated things, you will get things that are from one tissue or the other. You will not get it from both. And so most variegated plants, there are a few exceptions, will not come true to seed. And so just keeping in mind that then that asexual propagation is the best way to be propagating them. And I show this because, we tend to think of it in terms of plants. It's really popular with ornamentals, but it happens in all creatures. This is a chimeric cat. One cat's body, 2 completely different tissues. There's been a few of those like sensationalist TV shows with women who had womb tissue that was genetically different from the rest of their body. And so had babies that looked like they were their nieces or nephews instead of their children. It happens all across the board, but just knowing that this is going on. And also variegation, we typically talk about this green and white, but there are a whole bunch of other things that can happen as well in terms of different tissues in the same plant. So I talked a little bit how it affects propagation. Knowing where you're propagating from is going to be an issue. Also just recognizing that most variegation is unstable. You can see here on the right, there's this kind of all green branch coming out of a nice green and white maple. Typically, if you have variegated plants, you have to spend a bit of time kind of trying to cut off the parts that aren't true to what you want. Generally does not come true to seed. I think I have a photo of an exception later on. So just be aware of that. And typically doesn't come from adventitious buds. You can see a nice illustration here from a horticulture lecture at Texas A&M about depending on where you're getting different cuttings of the same leaf, you can end up with plants that are green, plants that are white, or plants that are variegated. And so kind of being aware of that if you're trying to propagate something that's variegated. Best way, since I say not from adventitious buds, is then using true buds. So when I talk about, cuttings where you have that little bump on the stem, rather than trying to have a bud come out of a leaf or somewhere unexpected. Juvenile versus mature tissue. One thing to always keep in mind is juvenile tissue tends to be easier to propagate than more mature tissue. That's why a lot of younger plants are easier to graft, easier to propagate. And sometimes it's kind of hard to see where juvenile tissue is. Typically, though, it's at the base of a plant. So when we think of trees that have been cut down and new shoots that are coming up, typically the juvenile tissue is closer to the base. And so those will be juvenile shoots and easier to propagate. These are some juvenile shoots coming up from a fig. Some plants, they have a really nice difference between juvenile and mature tissue. And so this is a Koa seedling I found when I was doing Koa work on Big Island. Came up from seed. You see kind of this more almost like mimosa or like halicoa foliage over here on the younger leaves. And then as it starts to turn mature, you get these phyllodes or kind of those sickle-shaped leaf-like structures. You can even see one here where there's kind of juvenile at the end. So something down here at the bottom is going to be easier to try to propagate from than something up here at the top. And a lot of times you'll see for folks who are working with things like established trees, if they are trying to take cuttings from them, they're trying to get those cuttings from the base if it's a hard to propagate tree. Also, the term rejuvenation, this had never clicked to me until I took a propagation class. That word juve in there is from like juvenile. And so you can make things juvenile again by sometimes putting it through tissue culture, inducing shoots from the juvenile area, other things like that, and make your life a little bit easier for a woodier plant. Basic technique of propagation is division. This is something you probably are familiar with. In the mainland, I used to do this with daylilies a lot, sedums, kalo. This is how we normally propagate it, right? We have the mom in the center there, and then all the keiki around there, called an oha, together. And we'll sit there, and when the keiki are established enough to go on their own, we pull them off and turn them into huli and plant them out. You have to know how your plant grows. Some plants, if you try to propagate or divide them in the wrong spot, you'll end up kind of creating a weak plant that can't really survive it. As I say right there. So you generally want something that you're going to split into roughly half if it's a large clump, or know, like with kalo, how established it has to be before you pull off those keiki. Stem cuttings, there's different types of them, but this is something you will hear a lot. This plant is propagable by stem cuttings. Different types of tissue come up a lot, and they'll say, you know, like for hardwood cuttings, do this, for softwood, do that, for herbaceous. Some plants will root no matter what. Some plants really need that particular stage, and it can not always be easy to find. So for instance, down here in the lower left, you'll see kind of this like darker bark. It looks a little thicker. I don't know, I tend to think it looks barkier. I'm not sure that's a real adjective. But you know, that's what we think of as hardwood. Here, we'll see that it looks a little bit lighter. Excuse me, it looks a little greener. That's softwood. It actually looks a little bit softer, right? And then herbaceous. And herbaceous is things like this, where it doesn't look woody at all, typically. A lot of herbs. I'll go into this in detail in the next few slides. So hardwood has the reputation of being the most difficult stage to propagate. Again, it's because it's typically more mature. You saw kind of that newer growth was more softwood, right? So the older, more mature tissue, you know, tends to just not produce buds or, you know, roots or anything else. There are some ways around this. One thing that I know apple growers on the mainland do is stool beds. This is where they'll take a plant here, they let it grow up pretty big, you know, so it's well established, has a really robust root system, and then they chop off the top. And then they cover that with soil or media, and what'll happen is a bunch of shoots will come out from the base. They will then go and clip those off of that main stump in the center, and they're already in the soil, and then they just have to kind of disentangle them. So this is kind of the way that they get around it. Air layering, which we'll talk about, sometimes works on some hardwoods, and sometimes playing around with it. Like, is it really hardwood, or is it semi-hard softwood, or hard semi-soft hardwood? All of these little gradations. So sometimes just playing around with something that's a little bit less or more woody might give you better results. But some plants, they only will be propagable at this stage, so it is worth knowing about. Softwood, again, I sometimes find this challenging to look at. Some plants are mostly softwood. Sometimes it's kind of seeing that New Year's growth or new season's growth, since we're here in the tropics, to kind of keep an eye out for. One technique here, especially as you're working with things like softwoods or with herbaceous cuttings, I mentioned water and kind of evaporation can be a big problem. You can either cover, or what a lot of commercial propagators will do is cut leaves in pieces. And then if you have something that's a little bit softer, it's less likely to be losing water through as much of the leaf, but it can retain some of that leaf to photosynthesize. So just as a quick aside here. Also removing some of the leaves and just keeping a handful of them. Herbaceous, I mentioned, also called stem tip. Most of the herbs that you'll see folks propagating, plectranthus, I think we also call that Greek oregano here, mint, basil, generally are herbaceous cuttings. Pretty easy to root. These are the ones you'll see a lot of folks rooting in water. The only thing is that they don't have as much of that kind of hard woody tissue that makes it difficult for a fungus to get inside. And so they tend to be a little bit easier to get rots, bacterial or fungal. So just being able to control that, you know, it's kind of a trade-off, right? Easier to propagate, more easy to get diseases. Air and soil layering. This is similar to kind of that stool bed thing I talked about a few slides back, and also similar to cuttings. What you're doing here is you're leaving the inner part of the cutting intact, and then you're cutting off the growing tissue, the cambium and all the rest around it. There's some controversy about whether you peel off the entire thing or whether you peel off a strip. Last time I checked the literature, you want to peel off everything, might vary plant to plant. When you're peeling something off like this is normally called girdling and is a bad thing. It's used sometimes to kill trees. But what happens here is if you protect that area that's girdled, especially the top part of it, you'll still have that water uptake that's really important, you know, to keep the plant hydrated and flowing. It won't have the hormone movement. And what happens is you'll have hormone pooling at the base, and that will induce root formation. There's different ways to kind of do this. Commercially, you'll see a lot of folks doing it with sphagnum moss and then wrapped in a plastic wrap and then maybe foil sometimes or other things to kind of keep light from getting in there. Light causes algae growth for one thing, but also you don't want to fry the plants. So we talked about solarizing earlier. It can also be much lower tech. This is someone who used an Easter egg and put some stuff in there. Does not appear to be rooting very well, but you know, you can use whatever is around essentially. I've seen folks who've used mud and leaf debris and had decent results and just wrapped around with a big leaf. So, you know, kind of the sky's the limit. There's also modified forms of air and soil layering. So this is like a soil layering, for instance, where you'll sit there and again do that kind of girdling of the stem, but then put it under the ground. And sometimes they don't even bother with the girdling. They'll just touch it to the ground. And then it'll form roots. Eventually you clip the stem that attaches it to the mother plant, and then you can pot that. This happens quite a bit with some plants. My grandmother's house, we had a blackberry plant that did this quite a bit. It was a thornless blackberry, so we encouraged it to do that. But you'll see this happening in nature as well. Leaf cuttings. This is really cool when it works. It does not work on all plants, unfortunately. Succulents, a lot of them do really well from this. This is from an Instagrammer. Oh, shoot. I don't think that's his name. I might have misattributed this. But who propagates a lot of succulents from leaf cuttings. Other plants that are well known for this are some types of begonias, not all. And what's happening here is you can sort of see there's a little incision, you cut across the veins, and at where those cuts are, you'll get that hormone pooling again, and you'll get those adventitious shoots. So again, this is a shoot, not where you would expect, coming right out of the middle of a leaf. Another one that does really well from leaf cuttings is Sansevieria, also known as snake plant. This quick story, I had a friend during my PhD who his house had a gas explosion and they lost everything, spent months cleaning up from it. The only living thing that survived the gas explosion was a piece this big of Sansevieria, and he put it into soil and it produced a shoot. It had been sitting out in the debris for, I think he said, a month or two before he found it. So leaf cuttings are amazing, but just keeping in mind some plants, it's not going to happen. But look it up. For some plants, it does work really well. Root cuttings, these I don't see this being used as much, but it is a go-to for a few plants. Ulu is one. Typically, though, the big thing is the plant has to kind of go through a growing cycle and sort of a dormancy period almost. So it's what we're looking for there then is it's putting those reserves into the roots, and then we'll draw them up later. And so what you're doing is cutting a piece of root, typically one that's easy to get to, hopefully near the soil surface. Those reserves are stored in there. And then sometimes there are naturally occurring buds. Other times it's those adventitious shoots, and it'll form those coming from it. Ulu plants, like I said, this is really common. Other plants, I don't hear it as much. I'm trying to think. Agapanthus, I think on the mainland, we propagate it that way. But keeping in mind, if you do encounter a plant that goes through this sort of cycle, give it a shot. Grafting. As I mentioned, this is a whole advanced training on its own, so I'm just going to talk a little bit about some of the fun things you can do with grafting. One thing is that it can be done sometimes across species. This has some really neat ways of kind of getting traits from one plant into another. If you've ever seen those horned melons they sell at grocery stores sometimes, not the tastiest of things. They're bright orange with kind of green pulp inside. However, the plants are really good at resisting nematodes. And if you graft a normal melon onto a seedling of that, you will get a plant that has the nematode resistance in the root stock and then has the nice tasting melons growing off the top. Not all grafts are compatible though, sometimes even in the same species. And what you'll see, this is a one I saw on Oahu, I think, is what's called grafting compatibility. Sometimes that happens within like a month, a year, sometimes it takes 25 years before the graft becomes incompatible and separates. Be aware of this, especially if you're grafting a large tree, say near your house. You don't want to discover 10, 15 years later that the graft was incompatible and falls over on your house, or heaven forbid, someone nearby. Tons of uses. I think I talked before about, if you have a girdled stem, you can graft seedlings or graft pieces of branch to connect the top and bottom together like that. You can use it also sometimes to improve growth. I know a grower on Oahu who grows eggplant into a tree by grafting onto another species of nightshade and gets this really nice growth, kind of like you see like the eggplants here in the bottom right. You can graft pretty far and get some really interesting things. This is top right is a product marketed as ketchup and fries, where you're growing both potatoes and tomatoes on the same plant. It's a potato plant that you graft a tomato onto the top. I have played around with this a little bit. My experience has been that's a lot of stress on a plant to be producing lots of fruit and lots of like tubers below. But it is doable, and this is one of the plants that they kind of document. You can make this yourself though, instead of paying the, I forget what it is, $40 a plant that they have. Grafting compatibility can be taken advantage of though too. Some people use this to dwarf plants. If there's incompatibility where it's not going to cause the graft to fail and fall off, but where it's going to cause it to just grow slower. Also, sometimes grafting onto another species that's closely related but has better disease resistance. This is done with tomatoes a lot. Some of those older heirloom varieties that taste great but don't have great disease resistance, you can graft onto something that is disease resistant and it'll transmit enough to boost. Art, yeah, you see a chair that's made out of it. I've seen all sorts of fun things. Sometimes you'll see those kind of woven branches of ficus, other things like that. At its most basic, grafting is just cutting one plant and sticking it into another plant. You're trying to line up those cell layers, like cambium layers. I'm not going to go too much in depth into the technique here. Tons of YouTube videos that demonstrate it better. As I mentioned, if you want to try grafting before an advanced training comes out, I recommend starting with hibiscus, just because it has a really good recovery rate. Also, though, be aware as you play around, there can be bad consequences. I read a medical report once of a woman who was trying to graft tomatoes onto Jimson weed or datura to get some cold tolerance to them. Worked really well. She got 2 plants that survived. One night fed her family tomatoes from it, had no problem. Next night fed her family tomatoes and they all had to have their stomachs pumped. It turns out she'd grafted one a little bit higher than the other. And in one case, the rootstock of the Jimson weed had started producing toxic chemicals and had transmitted those into the tomato fruit. So just kind of being aware with that as you play around with different things. But again, this is a whole lecture and series on its own. Tissue culture, or TC. This is something, again, you'll hear it abbreviated to in the trade. Also, you'll hear it called cloning, more often in things like newspapers, popular press, or micropropagation if you're looking into scientific literature, just that you know what folks are talking about when it comes up. It relies on a term called totipotency. And this is, again, sort of like we talk about with callus, I mentioned at the beginning of this, from wounding, or I think the closest thing we can kind of think of is stem cells. And this is the idea that any part of the plant can become a new shoot or root. What you're doing essentially is cleaning off the plant, sticking it into a media that has all the nutrients the plant needs, and then using those plant growth regulators to force it to form new shoots. The theoretical advantages of this are huge. There was an economist who worked out that if you kept up at the normal rate, you could take one orchid plant and propagate it into 1 billion plants in the course of a year. Most of the time, it doesn't work out that well. There's limitations of space, money, labor, everything else. But something to keep in mind is any plant that comes out of tissue culture is juvenile. This is a big deal if you have a plant that's difficult to grow otherwise. It also comes out with some other things, though, like it doesn't have the cuticle layer that protects the plant from drying out, so you have to treat them really delicately. I'll go through a few quick examples just because I love tissue culture. Another advantage of it is you can ship things internationally without restrictions. I've brought orchids back from Thailand without a problem because of this, because they can see if there's anything else growing in there. And if they see it growing in a flask, they know it wasn't collected from the wild. They know that you had to go through a lab to grow it this way, and therefore it's been ethically propagated. Some plants that you can't grow otherwise can be grown through tissue culture. Lilacs, for instance, there was a huge challenge growing them for a long time. They now grow a lot of them through tissue culture. Hostas, I think, are another one where tissue culture is really common. You'll see also, I've mentioned that international shipping thing. I've even seen traveling internationally, some things like here in the center, where they'll kind of put a funny colored gel and ship it that way, and then tourists can take it home. Sort of like we have the certified plants in the airports here. Another big promise of tissue culture is you can get rid of viruses. You have to be really good at it because you have to collect this very tiny area of the shoot there and propagate it. But essentially, the thing about viruses is they tend to be in the tissues below, but they're not in that brand new shoot. And so if you propagate from here, you get the plant that doesn't have a virus and you can grow it out that way. And this is them demonstrating that with sweet potatoes over here on the right. One thing to keep in mind, I bring this up in tissue culture, but it does happen in other forms of propagation. Whenever you deal with those adventitious shoots I mentioned, you get something that's called somaclonal variation or somaclonal mutations, some people will talk about. This is if you stress the plant a lot by forcing it to grow really fast, typically with plant growth regulators, sometimes the plant can't copy its DNA fast enough, and it'll get little breaks and tears. And when it tries to repair them, they end up with something different. So a bunch of us, I'm sure, are familiar with the chocolate orchid. This is the original form that was propagated. When they put it into tissue culture, they were trying to get a lot of them out into the market. They ended up with a whole bunch of different color forms coming out unintentionally because of those issues with the genes not quite lining up. So these are different color forms. They each have gotten their own names. People like them differently. And again, while I bring this up with tissue culture, if you're pushing, the plant sometimes, and if you have a chimera, like I mentioned in variegation, you can end up with things that do end up looking different from the mother plant. So just be aware of that. Sometimes it's a good mutation like these, sometimes it's a bad mutation, more often, sadly. Home tissue culture, this is something that I get questions about every year, so I put this slide in. It is doable. There's used to be a site I haven't checked recently called Kitchen Tissue Culture, where you can do it at home using something like an Instant Pot or a pressure cooker to sterilize things, and using kind of everyday available ingredients to make the gel. Agar flakes we can pick up from the market here, multivitamins, fertilizer, and then some sugar, essentially. The big thing with all of it is it's what's called an aseptic environment. You also hear this called a sterile environment. I had a professor who drilled it into me that a sterile environment means nothing will grow there. An aseptic environment means it's clean of anything else growing there. So this is the big kind of thing with tissue culture is you have to keep everything except that plant and you from growing in that environment. And so that means if you have any spores on you or anything else on the plant, that's probably going to grow faster than the plant itself. So you want to make sure you clean everything really well, and you're always really careful. We use something called a laminar flow hood where it's blowing clean air to remove everything. but I've seen folks use the plastic setup like this with gloves sticking in. I knew a guy who did a whole orchid breeding program through this setup, so it's totally doable. And there's tons of resources online, and I think I linked to a book at the end if you're interested. Oh my gosh, it's question time already. I know that was a bit of a whirlwind of asexual propagation. Any questions? I see there's a bunch in the chat. Take a look at those. And again, we're going to stick mostly to, as Russell pointed out, the propagation ones.
Hey, James, when you answer the question, will you repeat the question for anybody who's in person? Thank you. And can't see the chat.
Sure.
Thank you.
Yeah, good point. Adventitious, I can go back to the slide on that. Oh, sorry, the question was, what does adventitious mean? Sorry, a bit of a frog in my throat this morning. So when we're talking adventitious, this is a shoot or a root forming where it normally wouldn't. So, if we think of a plant, we know where we'll see a shoot coming out typically. It's like a space on the stem. An adventitious shoot is one forming somewhere else. So forming at the edge of a leaf, on a leaf, forming from a part of a stem where we don't see a bud. I'm saying it not because I'm going to quiz you on this necessarily, but because it's a term you'll see popping up in the literature. And like I said at the beginning, I want you to just kind of be aware of this for when you're looking through things. Yeah, a whole bunch of stuff for little fire ants. I have a tiny bit of experience, but we'll direct that towards when we have the entomology talk. Division on Anthurium. Yeah, that works pretty well. Division and cuttings. Anthuriums are, what's the word, semi-epiphytic, something like that. So you can take cuttings from them, you can divide them. They do pretty well from that. Do not do well from leaf cuttings. But yeah, for sure. Some plants propagated particular time of year, like rosemary and lavender. Very true. So there's a lot of things in propagation, and even more so in grafting, related to plant dormancy and when new growth starts. Grafting on the mainland, we typically do while the plants are dormant, but right as they're we're hoping they're going to get ready to break it, because we don't want the graft to have to sit there super long either. In the tropics, I think that's a little bit harder for us to tell sometimes, but the plants do go through these kind of cycles still, and just learning when those cycles are. Full disclosure, worked for Frank Sakia at Frankie's Nursery in Waimanalo for a while, and he is an expert grafter, and that's his trick, is he knows the cycles of plants really well, so he knows when the best time of year is for most of the plants to propagate them. And so, he'd ask me, I had a really good Suriname cherry I wanted to give him. He said, okay, take cuttings in the spring, please. And so for sure, knowing the plant and knowing kind of how it fruits, typically it'll be kind of coming out of dormancy and flowering a little bit after that. So if you can see when it flowers and fruits and kind of step backwards, that might be a better time to propagate. Sorry, oh man, one second. Ooh. Type of lavender, yeah, can make a difference. There's different species. Lavender is another one where water and moisture management is really important. Fungus can be an issue, so free draining, but still some moisture retention. Aloe is a rooting hormone. I have never heard that tried, but I think I've heard folks talking about it. I don't know that there's good documentation about it. Conversation about variegation. I'm not going to go over all of it again, but I'll go just real quickly to talk about, since I also brought up the term chimera again, I know that might be a less familiar term. When we're talking variegation, we're talking about two different types of cells that are genetically different in the same organism. And so because of that, you can sometimes have plants coming up that look very different. The plant stages regarding propagation timing. I think I talked about that a little bit with dormancy. It's really going to vary a lot with the plant you're interested in. I would look at what are the rainfall and season cycles of where the plant's native to is a good place to start. It's one thing we look at with orchids. Daylight, temperature can play a role. Water as well. Compatible plants for grafting. It really varies by the plant you're interested in. Pears, for instance, they have charts showing what's graft compatible, what's not. Tomatoes, pretty much any tomato is graft compatible into any other tomato. Experiments beyond that, you have to kind of play around. I recommend reading up on some of these and seeing what you can find. Yeah. Hibiscus, I haven't tried aloe, but as I mentioned, if you want to practice grafting, I really recommend hibiscus. You do have to keep the top covered, again, that moisture control, but it does really well. Kalani has a really good factor in the chat. I encourage you to read. I'll read it out. Sorry, I keep forgetting to read the questions. But Kalani says, some factors regarding stages. Does the plant have energy or carbohydrate reserves, or has it used them for recent flowering, fruiting, or leaf flush? Is there enough moisture in the plant so that the bark is slipping or easily removed to expose the cambium? Especially important for grafting. Does the cutting have new vegetative buds ready to pop, or does it have flower buds, etc.? Questions on spore propagation, mushrooms, ferns, mosses.
Talk about it, James.
Oh, okay, excellent.
If you're getting pretty overwhelmed with questions, so I guess Kalani, do you want to talk about it, or is that something you want to talk about at a later date or in a separate session?
I can just mention it real briefly. I mean, you know, it's so specialized, and there's just so many. This is such a huge topic. We have a whole semester course, TPSS 420, plant propagation, which you can take, and it wouldn't even cover necessarily all of these, but you know, ferns are a little specialized. There are some protocols you can look up. One thing I've learned from collecting ferns from the wild to propagate is that you have to get familiar with their physiology and know when the sori are present. That those are the spore-containing structures. When they're mature and just ready to open. So that's when you would maybe collect the frond, and then you'd want to wash it, rinse it off very carefully, because fern spores are so small that they're everywhere. And so if you probably have spores from other ferns on top of your fern frond that you're collecting. So you wash it off, dry it, put it in a paper bag, and let it mature. And then the spores will collect in that bag. Then you can sow them on a very fine potting or propagation media, or on agar or in tissue culture. And of course, that you would want it to be sterile if it was maybe sealed in tissue culture with nutrients. But you could probably do it on agar with no nutrients. Yeah, keep it moist. But that's just a real brief one. You know, that's a whole nother class to get into. And some ferns can be propagated vegetatively. You can take hapu'u logs. There's some ferns that have stipules, which are fleshy structures at the base of the frond that can be propagated vegetatively. So each one is different. That's my briefing. Yeah.
Oh, that's great, Kalani. Thank you. I don't have as much experience, so I'm really glad to hear from someone who does. And I would also say, if folks are interested in ferns, especially, unfortunately, Russell, I know that's coming from your group of folks. The two places I can recommend are NTBG over here on Kauai. I know they have a fern expert who does a lot of work with the native ferns. And then Lion Arboretum over on Oahu, I know does some work with them as well. Mushrooms, I know here we're talking about doing it for an advanced training, and Big Island does have Hamakua Mushroom Farm. Might be worth seeing if they'd be willing to do something like that. Yeah, they're a whole different world though. And I don't think I have a slide on them in the seed section next.
Yeah, we're thinking about the mushroom workshop, James. So we might cover that on our own later on. And I know that's kind of outside the realm of what we were talking about here. So thank you.
Sure. Yeah, and I guess the one last thing I'll say, and not to kind of throw him to the dogs, but Nu Wen, who's a professor at Manoa, is a real mushroom expert, and I know when he was a graduate student, led a mushroom cultivation club. I don't know how active he is now. He teaches a ridiculous amount of classes, but if he has any free time in the future, he might be worth talking to about that. Let's see. Problems with cloning, tissue culture, some genetic information is lost, higher chance of mutation. Yeah, so again, that's that somaclonal mutation I was talking about, right? And I guess the thing to keep in mind with that is it's almost always from forcing really fast growth, like faster than the plants can do outside of test tubes. And that's through really large hormone concentrations. Not something we typically have a problem with as much if we're doing normal propagation, except those chimeras that I talked about, the slide with the cat that has two different half faces. Media for cuttings varies a lot by plant. Yeah, so different plants like different things. I grew impatiens in just perlite with fertilizer for over a year without any problems. Other plants need something with a little bit more organic. Is that a real cat photo? That is a real photo. That is a real cat. Yeah, and there, like I said, there are humans like that too. Yeah, oh, okay, yeah. And great thing from Kalani in the chat, I'll read out for the folks in person. You can register or take courses at UH Manoa as an unclassified postgraduate student. CTAR TPSS 420 is plant propagation, taught by Orville Baldos, who we will hear from April 9th, his ornamental talk, typically in the spring semester. And he's a really great guy, really skilled in many things. Yeah, cats are usually only female if they're chimeric. I think that's most of the questions. If there's any last questions. Okay. Forcing growth with a hormone to practice, create. Oh, can you essentially asking if you can induce somaclonal variation in plants? There are folks who do this. There's something called mutation breeding also, where you can force mutations in plants. I wish I had a photo to show you. I did this once during my PhD. I treated 100 seeds, and I think 5 of them survived germination, and of those, 3 survived to maturity. It tends to be more bad mutations than good, but you can get some really interesting things. Fun fact, if you have ever eaten a stringless pea, that comes from an induced mutation from a chemical back in the 70s, I think, that was then integrated into other peas. Yeah. And yeah, it is sometimes also used for new ornamental things, but it's, you, like I said, you tend to get a lot more bad than good. All right, I know that was a whole bunch more information. I am going to go get some more tea since my voice seems to be failing me a little bit. Say, I like round numbers. Let's say 11. We'll all come back here. I'll come back a minute or two early if any more propagation related questions come up in the chat. But we are recording this.
Sorry, I was talking to my class. How long are we breaking for? Till 11? Yes. Yes, Russell?
All right, it's 2 minutes before 11. So I'm going to go through some more of the questions. We're not going to get back to the lecture for another minute or two. See some discussion about cat genetics. I cannot speak to that, but there are different ways that chimeras can kind of come up. And it can also be mutations that happen and then tissue that grows differently too. I talk about chimeras a lot in variegation, but I wanted to say that there are all sorts of other ones that happen. A lot of apple varieties that we grow came as a chimera, one that has nicer red skin growing on a tree that otherwise was kind of red with, or green with red dapples. Or I'm trying to remember the name of the tomato. There was a tomato where there was one branch on this guy's plant that had much larger fruits than the other branches. It turned out that it was a mutation. He saved seed from it, it came true, and could grow it from that. So yeah, there's all sorts of different mutations that can come up, sometimes in a growing plant or animal. But again, animals beyond the scope of this talk. Agar-based, can you use agar-based medium to root plants? The biggest issue with agar is it grows plants well, it grows bacteria and fungi even better. So if you're using it in tissue culture, you have to control the environment to prevent those bacteria and fungi from getting in on wind or on your hands or on the plants. So that's why you have to have kind of these controlled environments like a test tube or like kind of these glove boxes that I was showing you. It will, it's a very small kind of tangent, I will say it's used a lot now in cooking. It gives things a kind of nice gel. I saw a bunch of vegan cheese recipes that call for it, actually. Question about downloading the lecture slides and each week's lecture. Talk with your county coordinator. You have to sign up for CMS, which is through them, and then I think we're putting them up on there, but that'll vary a little bit. The videos, I think we're putting on a central playlist on YouTube that you can't search for, but if you have the link, you can watch. And if one of my fellow coordinators wants to share that, that would be great. Let's see. Milky sap plants need callus before planting or likely to rot. Yeah, and some plants, that's a good point. You know, some plants I mentioned, you don't want them to dry out, but some plants you do have to kind of have them dry out at the wound site to make sure that they're going to heal properly and then produce roots. Does it help to use rooting hormone on plants after they have calloused? It can. Again, some plants like it better than others. Some will kind of more naturally form roots. The rooting hormone is essentially sending the signal to form roots. So sometimes they'll just form that callus and it won't do anything. It'll just keep growing into this big lump of callus. If you have the rooting hormone, that's a way to kind of send it that signal. Saying, yeah, Chicago peace rose was a mutation of the peace rose. Yeah, exactly. And there's, I am sure right after this lecture ends, I will think of dozens of examples that don't come to mind. Like I said, there's bunches of them out there though, and ornamental trade is full of things where people found a mutation and then propagated that separately. I have a so question about propagating ulu. As I mentioned at the beginning of this talk, I am not going to go into specifics for any particular plants too much. I'm trying to squeeze in a bunch of concepts and terms because to take a semester's worth of content basically and put it into the course of a few hours.
Well, it's really, James, we got folks answering online through the chat, so it's kind of nice to have people answering through the chat as you're talking anyways a little bit, so yeah.
Awesome. Okay, yeah, and I would say for ulu and other plants, I have a list of links at the end of this thing when we do get the slides out to you that has native Hawaiian plants specifically, there's all this stuff about how to grow them. I haven't specifically looked for root cuttings, which I mentioned before, but I'm pretty sure one of those places lists it. And otherwise, check the CTAR website for the publications, because we do try to kind of put that stuff up. See a question from Russell about cutting a fallen hapu'u. I haven't actually done this. Kalani, I see you're setting up for kind of the hands-on aspect y'all are doing after this. Have you grown hapu'u from a fallen one? Do you want to comment on that?
We kind of got that answered.
Oh, okay. Yeah, okay, you guys got it?
No, go Kalani, sorry.
Oh no, I just know, like, you know, the nurseries, they'll export hapu'u logs, they just rip them out of the forest, and there's so much hapu'u on Big Island, but yeah, I mean, that's a tough one. You know, I think now with rapid ohia death, little fire ant, we don't want to do that. But yeah, you can just plant a log, you know, and take some time. You got to really keep it babied, you know, it has to be growing in a nice, cooler, cloudier, shadier environment, but you can grow hapu'u from spores. It's just very, 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 very slow. And we do have one person on Oahu. What's her name? Kay Lynch on Johnson Road in, is that Hakipu'u or Kualoa kind of area? So look her up.
Thanks, Kalani. Russell, any other questions from your side that you want to?
No, that's it.
Okay. And again, yes, slides of this will be available. Recording of this will be up. Give us a little bit of time, especially for the recording. YouTube has to process it, and then we have to go through and cut out, so you don't have to watch a 10-minute blank section of it. Yeah, all right, I'm gonna close the chat and move on. Oh, and no ill feelings about this cartoon. I just saw it about plant propagation and thought it was a fun one to stick in there. Let's see. Okay. Sexual propagation. I swear I did not steal this picture from Russell. We just both coincidentally happened to have it in our presentations. And I think it speaks to what a good illustration it is of plant parts. I'm not going to go into detail of it, but if you hear these different terms being talked about, again, that's what I want you to take away from this talk, is these ideas of different parts of the plants. So things like the stamen, you'll hear folks talk about, consists of the anther and the filament, is the male, quote unquote, parts of the plant. The carpel, which consists of the stigma, style, and ovary, is the quote unquote female parts. There's all sorts of different combinations and variations with these. As Russell had mentioned last week, orchids have all sort of combined into a column with that then has pollinia. But if you're looking to pollinate a plant, which comes up in a lot of sexual propagation stuff, this is the parts you want to know where they are and how you can kind of control them. So seed germination, I'll talk about first. This is another thing. There is a whole class on this at UH Manoa, again, taught by Orville Baldos. So one slide encompassing a semester's worth of class, right? It's nationally regulated, I guess, is the one thing to really take away from this. Anyone who's selling seed has to be testing their seed to say what the percent germination is, percent purity, and looking for other objects in it. Stones are a really common one. Seeds from other plants is really common as well. Certain plants, there's a higher threshold of that than others. And also seeds of different cultivars. So Russell talked about that before, right? You know, if you buy a packet of blue morning glory and you plant it and there's a few red ones in there, well, you might be happy, but you know, that's not what you paid for. And I had that happen once. I bought a whole bunch of pumpkins. We were doing a fundraiser when I was a grad student, and we were going to grow up all these pumpkins and sell them at Halloween. Well, the seed company had a mix-up, and it accidentally put in spaghetti squash. And so we ended up with this huge field of spaghetti squash. And people are not as excited to buy spaghetti squash for Halloween. So this is what this kind of national regulation is geared towards, to prevent those sorts of things. Sometimes it works, sometimes it doesn't. All commercial seeds have to have the percent germination listed on there. It doesn't have to be at a certain number generally, though. I think I may have said this last week, entirely possible, that I bought seed once that said 15% germination. And as long as they list that, it's still saleable. And they also generally have to list when it was packed, so you'll know how much it might have degraded. There's different ways of testing germination that they do. One that's kind of approachable for us is they use paper. It looks just like kind of the normal paper towels that we use, but it's a little bit thicker, absorbs water differently. We'll put a certain number of seeds on there and then count how many germinate. And I used to do this when I worked for a company before, where you just roll it out, leave it for a week at the right temperature, and then do that. There's another one that's a chemical treatment, TTC, and I always forget what that stands for, where they dye the seeds, essentially. It does kill them, so it's not the ideal way to do it if you're working with a limited number of seeds. But you can see down here in the bottom is seeds treated with it. The ones that are dead don't get dyed. The ones that are alive do get dyed and die, ironically. And then if you have a mixture, you can kind of count the percent of alive and use that for ones that would germinate. So these are different sorts of ways that the seed industry goes on, just so you kind of know what's happening when you see that on a package. Seed dormancy is a really big challenge for some plants, less of a challenge with some tropicals, just because, again, we're growing all year round. But there are different sorts of mechanisms for it. There's seeds, I'll talk about in the next few slides, that require some sort of physical barrier to be removed. There's seeds that require a chemical barrier, like these two, which I'll talk about a little bit. And then there's also ones where it's kind of a growth pattern. They have to have a certain amount of cold or some other situation. Excuse me, here I have tomato and then papaya seeds, because these are ones that a lot of folks do seed saving for. Both of these seeds, when you think about them when you're eating them, they have that kind of gelatinous coating on the outside of the seed. That coating contains, among other things, some chemicals produced by the plant that inhibit it from germinating. And this is a way to kind of let the seeds get further away, theoretically. The plant doesn't want all of its seeds to come out right below it, because then if something happens to that area, all its children are gone, right? So it wants it to be spread. So typically this can happen a bunch of different ways. The way that we do this a lot of times is through fermentation, not long-term fermentation, but just like a few days. You can see there's kind of a white gross film happening over on the tomatoes here. This is what I do. I have one I'm doing in my kitchen right now. Not the best smell, but what happens is if you leave it just for like a day or two, the bacteria and fungi will break down the pulp and will break down that kind of gelatinous coating and will break down those chemicals that prevent it from germinating. The challenge, though, is you don't want to leave it too long, otherwise they will start germinating all in the bottom of that jar, and then you'll have a bunch of germinated tomato seedlings that you have to deal with. You want to get them while they're seeds and before they start germinating, before they start swelling up and having that root come out. And then what you would do with this is pour off the top, add more water, and slowly kind of keep pouring off the pulp. And I think, this is something that you can get demonstrated during your hands-on events for this. But it's a pretty easy process once you get going. I do this also for other seeds that have a gelatinous coating. I've done this for papayas before. One sort of thing I will share with that is best to do if you have somewhere that's not where you're also preparing food, so that these fermenting tomatoes and papayas aren't kind of contributing nasty odors to your food as it's going on. But that's one way to remove some forms of this seed dormancy. Another one that comes up is scarification. Scarification, fancy word for cutting. The way this evolved, just so you kind of have the background for it, is typically by being eaten by an animal, and so it's to survive their digestive system. I talked before, last slide about they want to get it far away from the parent plant, so it has a better chance of surviving. So this, in some systems, like you think of a bird, for instance, like this wonderful chicken diagram, it goes into the crop. The crop of a chicken also has bits of rock, other debris that grinds the outside of the seed and breaks that seed coat. And so then the seed coat can let water, the water can get through the area where the seed coat was, and then the seed can germinate. It's also used as a way for weeds, though, to survive in the soil. Some of you all, I'm sure, have had the experience where you're planting an area, you've weeded it really well, you turn over the soil, and suddenly new weeds are coming up. No, they didn't just magically appear, they didn't all blow in. A lot of them have this kind of thick seed coat that allows them to sit in the soil for a really long time. And what happens is there's different grades of bacteria and fungi in the soil. And so as they get moved into these different layers, different bacteria and fungi digest the seed coat. And when they're up near the top, normally it's enough that it breaks it, and then they can germinate. But it's not only in weeds, it's in a lot of other things. There was a question earlier about Koa. This is the way we germinate Koa. I mentioned nail clippers. File is another way. Knife works really well. Please, please, please watch your fingers if you do that. Sandpaper is another really common one, putting it between two pieces. I worked with a wild legume for a while where we would do it, and you would rub it between the sandpaper and you would hear just this very light pop. And that was to know that the seed coat had broken. Some it's, it has to be a lot. Some it doesn't have to be too much. There's also chemical ways to treat them. In the olden days, they would use sulfuric acid. I was in a lab once where we did that for treating tree seeds, soaking them in sulfuric acid for an hour, I think it was, something ungodly like that. Not something that's fun to handle, though. So, be careful if you do. So other folks have used other types of acids. Some seeds, again, I'm not going to go into every single type just because there's so many. Boiling water is really common. Some people think this might be simulating kind of a forest fire type event. So make sure, as you're looking for different sorts of plants, thinking about, okay, does this need a seed treatment? And if so, what is it? One thing, hibiscus, for instance, a lot of hibiscus relatives need to have the seed coat broken up in some degree to germinate. So kind of keep that in mind if you're working with different seeds. Stratification is another term you'll hear besides scarification. This is a combination of it being cool, but also moist. And the reason I emphasize moist here is, besides my whole thing about moisture being an important thing for plant propagation, I've seen a lot of people who think that by storing seeds in their refrigerator, like in the packets, that they're stratifying their seeds, and they're not. You need to have that combination of the coolness and the moisture. And this is kind of envisioning like an overwintering experience. So again, as I put up here, not something you always see in tropical species since we don't have that strong a winter effect. But if you're trying to grow some temperate plants here, some of them will need that to germinate. Apples, persimmons, the list goes on and on. The reason that the term stratification came out, because a lot of people hear that and they're like, what does that have to do with, cool and wet? Is because the olden days, the way that this was done was layers of different kinds of media. So like here is the illustration, gravel, sand, then the seeds, then more sand, then gravel. And so it's strata, layers, that were then cooled and moistened. And so, even in areas with winter, they'll put them outside and do it. I knew someone who would sit there and, much to his husband's chagrin, would use a whole vegetable drawer of the refrigerator for this. And I, at one point in my PhD, much to my lab mate's chagrin, took over a refrigerator and left the light on all the time. That is something also to think about with stratification. Sometimes they need that light to signal germination in addition to being cool and moist. Sometimes they don't. And just knowing that about the plant that you're looking at. Also, again, I've brought this up so many times, but with moisture, you want to make sure that you're also not keeping it too moist, because then you're going to suffocate the seed, but also you're going to, if it's too moist, get things like fungi and bacteria growing in there. Something I've seen, not 100% sure about the science in terms of how it holds up, is folks adding a little bit of hydrogen peroxide with the water when they're soaking the seeds. And then that kind of helps kill some of the stuff. Might not work as well if you're using organic things to kind of keep it cool and moist, because then it's interacting with the materials in that. but just kind of an idea that I've seen floated around before. Seed saving. I will start out before I get into this by saying that we're currently planning to have an advanced training statewide from another group I'm involved in, hopefully in April. We're gearing it not towards the general public, but towards folks like you all who have kind of a little bit more in-depth background. So it'll be master gardeners, I think Go Farm, and I'm trying to think there's like one or two other things. So we'll go way into depth on it, but I'll touch on it here a little bit. And I'd like, if any of you do practice seed saving now, let me know in the chat, or you can kind of do a raise hand emoji. Oh yeah, I see Laurel's doing that, a few other folks. Yeah, I got this picture because I really liked, showing all the different seeds and all of that. But as I will talk about in a few slides, what really bothers me about it is that this is not the right stage to save seeds from two of these crops, the ones that I've circled. If you save seeds from that green pepper, I can guarantee you are going to be setting yourself up for heartbreak. Same thing with that eggplant. Other ones on here, fine. And I'll talk about that more in a few slides. Yeah, okay, yes, seeing tons of stuff in the chat about it. Excellent. Beautiful, yes, okay, I'm so glad. Oh, and I see a question or a comment about hybrids. I will talk about that in a few slides also. All right. So again, trying to get into kind of some stuff about what we're doing when we're thinking of seed saving. I want to kind of give you the background so that you look into, when you look into the specifics, you'll know what they're talking about. Things to keep in mind: health of the plant. Again, I would not want to save seed off of this plant. Some viruses aren't transmitted from seed, some viruses are, but again, you always want to kind of pick the best one you have. Is the plant true to type? If you want white, white flowered impatiens to keep growing, and you save seed from the red one, chances are you're not going to get that white one. On the other hand, if you have a bunch of white flowered impatiens and you have one that the flowers are twice the size of the others, maybe you don't want the one that's normal, you want that one that stands out. So just kind of being aware of what your own goals are with the seed saving. And then, as I mentioned in the last slide, making sure the plant is at the right maturity. The right maturity is not always the stage we eat it at. There are peppers that are green at maturity. They're not super common in the trade, which is why I pointed out on that on the last slide. Eggplants are one, though, that we don't eat at maturity. When they're that beautiful kind of purple or green or striped color, they're immature. Most people don't like the kind of fully developed seeds that are a little bit harder. When an eggplant is mature, it's typically more like this bottom one where it's sort of yellowish brown. Cucumbers are another one. Cucumbers tend to turn yellow, brown, sometimes scaly, and so look very different. And just kind of learning that about whatever plant you're saving seeds from, what's the right stage to save the seed at. It might be what you eat it at, but it might be months afterwards. All right, hybrids. And I saw someone had mentioned this in the chat earlier. Again, I'm not going to give you all there is to know about hybrids. There were whole courses on this. If you take nothing else away from my talk about hybrids, take away this first point. A hybrid is not a GMO. Can someone tell me in the chat what GMO stands for? Because I know there's all these gotcha YouTube videos about it. Genetically modified org. All right, go to the last step. What does org stand for? Organism, thank you. Yes, GMO stands for genetically modified organism. Very good. A hybrid can be a GMO. Not all GMOs are hybrids, and not all hybrids are GMOs. These are kind of independent things. A hybrid is an offspring, and this is the definition we're working with here, of two different cultivars. You remember we talked about cultivars, or sometimes we'll call them varieties, or sometimes two different species. The big thing, and I think this was brought up in the chat before, is they're often not true from seed. And so this is a reason that people don't like to save seed from hybrids most of the time, is because you don't get the same plant that you're saving seed from. This is used a lot, though, in the trade, and sometimes it's used as a way of kind of controlling it so that you can't save seed and get what you had. You can still save the seed, you're just going to end up with something different. But it also relies sometimes on what's known as hybrid vigor. These are kind of two different terms, hybrid vigor and inbreeding depression. I didn't put them up here. I'll try to remember put it for the next time. Hybrid vigor is the idea that if you take two very unrelated things and put them and cross them together to make a hybrid, you get something that's stronger and better than either parent. This is true in corn. It's not true in all crops, but here you can see kind of one parent, the other parent, they're both sort of short. Cross them, you get the super vigorous plant in the middle. And same thing with their corn cobs in terms of number of seeds, growth, all of that. Inbreeding depression is the opposite. And I think I'll talk about it a little bit in future slides, where if you're inbreeding some plants, you get all the cliches we see in movies like Deliverance or things like that, where the plant is less vigorous because of it. So keeping that kind of in mind, hybrids, you can take advantage of that though, and get that hybrid vigor in your own plants by making your own hybrids. And you can save seeds from hybrids and select out plants from that. It might take a few generations of saving seeds, but you can get something that's stable also. I had to put this in just as it's one of my favorite images I've ever made, and I never had a chance to publish it. So I'm living vicariously through you all sharing it this way. This was a cross I made between two species of pea. On the left is kind of what we see as a normal type of pea, and then across is a different species, not just a different cultivar, but a different species, a wild pea. And this is the one I was talking earlier also where I had to rub to break the seed coat. You cross them together. This white pea was, maybe 2-3 feet tall. The other parent, very spindly and maybe 3-4 feet tall. The F1, the hybrid between them, was 8 feet tall, something like that, I think. It grew off the bench up into the lights and eventually burned itself on the lights in the greenhouse, but kept producing. That's that hybrid vigor that I'm talking about. I bring up the term F1. It stands for filial one. You will almost never hear anyone say that, but you will see F1 in catalogs a lot, especially when they're talking about hybrids. If you save the seeds from an F1, you're going to get what's called an F2. And so this is assuming that you can kind of inbreed them, or you can cross the two hybrids that are produced here together. And you can see the variation here in these peas in terms of color, in terms of kind of flower shape, all sorts of differences with that. And that's really typical. If you save seed from a hybrid, you might not get this range, but you're going to get some range of diversity. It might be different heights, it might be different shapes, might be different flavors even, but all sorts of different things coming out. So you can save seed from hybrids, but just be aware of kind of what you might be getting from it. That's kind of my takeaway on hybrids here. Let's see, anything else? No, okay. Oh, sorry, and before I move on, F2 is what this is normally called, again, filial 2. You will also once in a while see BC or back cross. That's when you take a hybrid and cross it back to one of its parents. So these are just terms, again, I'm not going to be asking you about this, but you should be aware of these terms for if you're looking into the literature, especially with plants with pedigrees. This comes up really often, back cross to this parent. And that's a way of getting more genes from one parent into the same background. Things we can't do in animals, especially not humans. All right, let's see. Seed saving crosses, how to make hybrids. This is going to vary a lot plant by plant. And I think I talked the next slide about inbreeders versus outcrossers. The main thing that you're trying to do is to only get the pollen you want onto that plant. And that typically means knowing how your plant is pollinated. Some plants are pollinated by wind, some plants are pollinated by insects, it's some combination. There are plants that are pollinated by, mice, earthworms, ants, the list goes on and on. And so what you want to do then is control whatever else would be doing that pollination. We typically end up closing things. I'm using squash because it's really easy to see. It's not small sort of flowers. And what we would do is the day before they open, you'll see the flowers kind of turn color, but it isn't open yet. We would close it. There's all sorts of fancy things you can close it with. We normally would close them with like these little ties that you see to hold together produce, or twist ties on bags. Excuse me. The other thing folks will do, and you'll see this term a lot in the literature, so I want you to kind of be aware of it, is emasculation. We use this colloquially for a whole different meaning. In this case, scientifically, what we're talking about is removing the male parts of the plant. And that's so that they're not going to be contributing to it. In plants like squash, that's less of an issue because they're separate from the female parts. I know Russell talked about this a little bit. In some plants, though, you have the male parts and the female parts in the same flower. And if you don't want that plant to self-pollinate, some plants can, some plants can't, but you have to control those male parts. And so the easiest way sometimes is to remove them and just leave that female part. So then you can go and stick the male pollen onto it. I know this is all super hypothetical and can be a little hard to visualize. But again, I want you to just sort of have an understanding. So when you see it popping up again, you know what they're talking about. One thing to kind of keep in mind is identifying the female and male parts. I showed you kind of that diagram at the beginning of this section. Some plants make it really easy. On a squash plant, this is what a female flower looks like. It has a little miniature squash at the base. And yes, that is the exact shape that the mature squash will have too. Male flowers don't have that, and they also have pollen. Learn with your plant what it is. If you can't find things for that plant, look into relatives of that plant. You can type into Google, what is cucumber related to? And it'll tell you squash, and you'll know, okay, roughly this is what I'll do. So you take the pollen from the other variety you want, or from, if you're trying to save seed and want to make sure it's ultra pure and nothing else is getting in there, you take the one from that same plant and put it on there. You basically are just trying to control who's doing the pollination. So it's you and not the insects or the wind. Then you close it up again so nothing else can come in and mess up all your hard work. And you wait for the seeds to develop till it's at that right maturity stage. So I bring this up partially if you wanted to make your own hybrids. It's not going to be a large number of people here. But also if you, like I said, want to save seeds and you're growing something else nearby that might contaminate it. Or your neighbor is growing something nearby, or your research station is growing something nearby, that you can kind of look into how to control this. Okay, I talked about these terms a little bit in earlier slides, so I'll go into a little bit more detail here. You'll hear about outcrossing and inbreeding. This, again, plants function very differently from animals with this. There are a lot of things that we cannot do in animals that are very common in plants. Inbreeding is a plant that doesn't mind being inbred. Tomatoes are a great example. Peppers, I'm trying to think of a few others. Wheat. Yeah, all of these are plants that typically we've domesticated so that they're fine pollinating themselves. And because of that, they don't experience that thing I was talking about a few slides ago, inbreeding depression. There are some differences too. Wild species don't tend to be inbreeding. Domesticated species, it's a lot more common. The reason I have this particular tomato photo up here is because this is what a cultivated tomato looks like, and this is what a wild tomato looks like. I know this is hard to see, so just keep this in mind when you get this slide to look at on your own. But those of you who can see it, there's a little bit of green, that stigma sticking out of the wild tomato. That's because this one wants to be pollinated by something else. This is kind of what the normal wild species relationship is. We, as humans don't want that. We don't want, this nice tasty tomato we've selected to cross with this bitter tasting awful tomato that grows wild nearby. We want to know what we're getting from our seeds. So we have unintentionally selected for ones where the stigma doesn't stick out. And so then it pollinates itself and you don't have that problem. So this is when we're talking about inbreeding, what's going on. As I said, normally this is something humans have selected for, but just being aware of this so that sometimes, if you have an inbreeding plant, you don't have any neighbors or anyone else growing anything nearby, you can save seed from that without having to go through the steps I talked about before. Outcrossers, on the other hand, do not like being inbred. They do not respond well from it, and you get that kind of inbreeding depression I was talking about, where if you're inbreeding them, you end up with weak plants. You can sometimes recover some of that virility or vitality through crossing with other things that are kind of closely related. But the biggest thing with this is just, if you're thinking of seed saving, making sure that you have enough plants that are closely related enough that it's not going to result in inbreeding depression, but not so closely related that you have this problem popping up. And so this is really common, say, for corn. You know, when you see seed saving guides for tomatoes, they'll say you need 1 plant. For corn, you'll see you need 100 plants. And that's because they don't like being inbred. They need to have that exchange among the different plants. They might all look the same, but there's some hidden genetics inside that there's variation in. And that's what you're trying to keep that variation going. If you can't do it, I'll list a book at the end called Breed Your Own Vegetable Varieties. Amazing, amazing book. One of my all-time favorites. And she talks about something that I'd never considered before, which is if you can't grow 100 corn plants, which not all of us have that space, myself included, you can also do it over time. So maybe you save seed from 10 plants 1 year, and then mix that with seed from another group. And every year save from another group of 10, not from the one from the year before, and then mix them up. So you're kind of constantly adding back in a little bit of genetic variation. Again, you know, whole courses can be taught about this, but want to kind of roughly give you these concepts. Last sort of thing to talk about with this is incompatibility. There are plants that dislike being inbred so much, they have in fact evolved ways to make sure they do not get inbred, and not just kind of what I showed with the tomato flower. These are typically genetic, and they will actually cut off the pollen that's growing on the flower. They end up forming a kind of plug of that callus I talked about to keep the pollen from growing if they recognize it as coming from the same plant. Things like broccoli, other sorts of cole crops, that's really common. You cannot self-pollinate broccoli. It will not let you. There are ways to bypass it. They are not something I'm going to go into tons of details on. Some plants are more strict about it than others. But just being aware of that also, you know, if you want to play around and kind of push the barriers with self-compatibility or with outcrossing, just be aware that there's some obstacles you might come up against. And so look into that as well. All right, lots of information, I know. So seed treatments. This is something I bring up, not because this is not necessarily something you're going to get tons of information about or see a lot of, but something so you know roughly in case you end up with this kind of neon colored seed. The reason this started out was to reduce the growth of especially fungi on seeds. And I know that's, you know, something that we don't tend to think about. Is this a huge problem? The biggest crop I know this is really that has benefited from this is wheat. And so in the bottom right corner here is a harvester harvesting wheat in the Pacific Northwest. Those black clouds you see are not smoke. They are actually spores from a fungus that infects wheat seeds. And then while it's growing, and replaces all the seeds with clusters of fungi. So when the harvester is going through, it releases these fungi, they spread around, and then infect the new seeds as they're growing up. The way this was controlled, because you can lose up to 70% of your crop this way, was by adding seed treatments, where you're getting some sort of fungicide on the outside of the seed to prevent the fungus from growing while that seedling's developing and is kind of weak. I should also add, I worked with this fungus during my master's. In addition, to add insult to injury, not only does it reduce yield, it smells like rotting fish and is highly flammable. And so harvesters would catch on fire from these spore clouds as well. Anyway, so you see treatments with fungicides as a really common form of seed treatment. Legally, they are required, any sort of these seed treatments, to dye the seeds. It can be different types of dye. There are organic seed treatments involving fungicides, but they have to show that the seed has been treated with something. And so you'll occasionally see seeds offered in catalogs. I bought some this past year that arrived bright neon red like that. If you're working in organic fields, you cannot use treated seed if it's a non-organic treatment. You can save seed in a non-organic field and then grow it from that. But look into some of the organic regulations. I don't want any of you to kind of run up against that if you're organically certified. Another form of seed treatment, slightly related, but I thought I'd kind of stick it here, is it's really common also to inoculate legumes with nitrogen-fixing bacteria. Some of y'all might have heard that things like peas, beans, other legumes can produce their own nitrogen. The way they do this is by forming a symbiosis with bacteria on the roots. And that's a whole other lecture too. But you need to introduce those bacteria. Some places have them. They've already been growing there for a while. Some places don't. There's some difference of opinion about how wide a range of species these bacteria work with. Some people say, you know, okay, peas need one type of bacteria, fava beans need another, koa needs a different kind. And so you'll see folks who are growing commercially sometimes just say, okay, just in case, I'm going to treat it with this. If you've been growing the same legume there for a while, chances are there might be some of those bacteria there already. If not, you can buy the inoculum to treat the seeds with. One thing being where we are, there are a lot of restrictions on bringing in this inoculum. So it might be easier to buy it from a local shop than it is to try to bring it in your own. Or you could try and, you know, hope you don't get caught. I do not recommend that, both as a university employee and as someone who cares about the soil microbiome here. But just being aware of that as you're seeing legume seeds. And this is a whole different form of seed treatment. Seeds do not normally arrive pre-treated. There are other types of seed treatment that are a little bit more experimental. I'm not going to go into them here, but just kind of be aware of it as you hear these things brought up by different folks or different companies. All right, storing seeds. This is one that comes up, and this is something I still have to remind myself about. Seeds have very specific conditions they want to be stored in. There are seeds you can't store. The one that comes to mind for me most often is chayote. For any of you who've grown chayote from seed, you see it just starts popping right out of there. It's what's called viviparous, where the seed is actually alive and growing in the mature fruit. And so this is something where you have to kind of keep it going. You can't really store it. There are ways to store it in tissue culture, but you know, again, that's a little outside the scope of this. Most seeds like to be stored in cool and dry conditions. You remember I talked about stratification, cool and moist. So you don't want to accidentally be germinating all your seed. You want to keep them dry. And a lot of seeds, it varies, but it's roughly 30% moisture is what you want. That's hard for most of us to measure. So you'll see a lot of things about this many hours in a dehydrator or in a very low oven. You also don't want to cook your seeds when you're drying them out though. So you really have to kind of play around with this. Typically, especially in our humid environments, unless you're maybe on a leeward side, but even then, you're going to have a hard time drying it enough in the ambient conditions. You can dry it most of the way though, and then put in what's called a desiccant. And these are little beads. Sometimes they're made from silicone, sometimes they're made from other things that soak up that residual moisture. The big thing with this is you have to have something that breathes that moisture can get through. So I know I personally save a lot of seeds. I normally have them on my desk in those little bags, kind of like the ones you'll sometimes see seeds being sold in. Those are great for storage, but you have to make sure your seeds are dry enough to put them in there. So it's good to store them first in something like paper. Coin envelopes are really common. And then dry them out a little bit more in that. And then for longer term, put them in the plastic. And the nice thing about the plastic is it doesn't let moisture out, but it also doesn't let it in, so they can store a lot longer. A lot of people will store things in the fridge. This works really well. Some seeds don't like being that cool, but a lot of seeds do fine. I knew someone when I was at Washington State who had saved seeds from a tomato in 1982, had a big jar just filled with the seeds in her fridge. It's sealed, so there wasn't any moisture going in or out. And she just would take a scoop of it every year and grow them up. And it was doing that in, I think it was 2009 when I met her, and the seeds still had 80% germination. So they can last a really long time. Some seeds last longer than others. Onion seeds, for instance, are notoriously short-lived, 1 year, maybe 2. But there's different ways you can play around with it. One thing I will say, if you choose to store in a freezer or fridge, is a lot of them that are frost-free, the way they do that is by sucking out moisture. And so if you don't have something sealed and you have your seeds in there, it will suck the moisture out of the seed. While they need to be somewhat dry, they don't want to be completely dry because they're still kind of slowly breathing. And so you have to make sure then that you have something to prevent the moisture from escaping. So for instance, drying them down, then putting them in a sealed glass jar. Also, as kind of a last fun aside, have any of y'all heard of the Doomsday Seed Bank in Svalbard? Yeah, I see a few nods. So this was a thing that was made, gosh, I forget how many years ago at this point, where they went and looked into if there was some world catastrophe, where could we store seeds in these kind of cool, dry conditions I'm talking about, so that people could go and reclaim them later. And it's also ended up being a big thing for areas that are war-torn and want to save their seeds. So it's in Svalbard, which is an island up in the Nordic Circle, or in the Arctic Circle. I think it belongs to Norway. I could be wrong about that. It's run internationally, and countries can put it in kind of in a black box so that other people don't know what's there. But they tried to get the word out so that if there is some catastrophe, people will know, go up to this island in the Arctic Circle, and there will be lots of seeds there. Native American tribes have done a lot of seed saving and put things in there. As I mentioned, war-torn areas, quite a few countries have saved seed that way, so it didn't get lost during looting. But it's a really cool concept, and they're kind of taking advantage of these conditions I talked about. A few exceptions to seed saving, and this is kind of my last slide for this before I start going into kind of the last other resources and topics. These are not ones you will come across a lot, but they are worth mentioning. Polyembryony, this is particularly worth mentioning if you are trying to save seeds from citrus or mango, as both of these do that a lot. In this case, like in this citrus you'll see over here on the right, there's a lot of little embryos popping up from one seed. And what's happening there is it's actually one true embryo that came from the flower being pollinated, and the rest of those embryos that are coming up are from the actual mother plant, and so they're clones of the mother. It's not in all citrus, but a lot of citrus have this, and it's kind of hard to tell sometimes which embryo is the one that's from the sexual pollination and which ones are just clones. And so if you're trying to develop a new variety of citrus, this might be a challenge. You can kind of look and see if one shoot looks different from the others, but it's not always easy to tell. On the other hand, if you want to clone a citrus plant from a seed, this is really great. Mangoes, same thing. Apomixis is similar to this, but a little bit different. This is where the entire seed is replaced with a clone of the mother plant. Not something we see a lot of, but it's worth bringing up that dandelions, for instance, the invasive ones, are typically do this. Sometimes a plant can do both, where it'll be apomictic on one flower and then sexual on the other one. But it's a way to, if there's a plant that does really well, it can essentially clone itself and spread that way then. So just being aware of these terms as you hear about it. And like I said, polyembryony is one that tropical plants come up with. I talked about variegation and how it's from this chimera, these two different tissues that are genetically different in the same plant. Most types of that, because you're only getting offspring that are from one type or the other, are not going to end up with that nice variegation. And of course, there are exceptions to this rule, and I had to put it in. This past year, I grew for the first time a tomato variety called Variegated, aptly enough, or also I think called Splash of Cream, that comes variegated from seed. It's not really exceptional as a tomato beyond that. It's decent, but it's really pretty. And as someone who does ornamental plants as a focus, it was kind of fun having this tomato plant that has these white streaks on it. I don't know the whole mechanism behind it, but it does come true from seed. And so just kind of being aware that, I say these things, but always look into it a little bit more for the plants that you're interested in. All right, I know there's a bunch of questions. We'll save those for the end, but I'm going to do a quick group quiz, everyone's favorite. Put your answers in the chat box, and I'm going to shift past the questions, and then I will go into these. So, wrong way. First question, what is arguably the most important environmental factor for propagation? Moisture, beautiful. See a few people answering that. Thank you. Moisture control also, yeah. Okay, why should you always read the label for chemicals you use in the garden? It's the law. Perfect. Yes. Legality. Yes. And your safety, environmental impact. Yes. Beautiful. All right. What are good things to look for in the plant you're propagating? I see some folks had answered that already. I saw one about, I think that came up earlier with the moisture question about plant health. Beautiful vigor. Yep. No viruses. Sorry, the question I saw asked for a repeat. What are good things to look for in the plant you're propagating? True to type, beautiful, age, all these things. What are adventitious shoots or roots? And I know I mentioned this once, and I think I circled back to it. I talked a little bit about how they're good. I don't know if I emphasize enough on how they can be bad. Roots forming where they normally don't, beautiful, yeah. Shoots, same thing. Ways that they're good, is that you don't always have to get a cutting or you don't have to always take a cutting where there's a bud. Ways that they're bad, and I don't think I clarified this enough, and that's my bad. You're more likely to have mutations if you have adventitious shoots than if you have normal shoots. So I talked about that with the somaclonal variation for tissue culture. This also happens for adventitious shoots. Yep. I see some other great answers coming up. Excellent. And finally, the last question. Oh, please, please, please answer this correctly. Are hybrids the same as GMOs? Thank you. No. Oh, I feel so validated. This is wonderful. Yeah. So many no's. Wonderful. Yes, it is possible that a hybrid can be a GMO and vice versa. They are not the same thing. And you will have saved me so many questions that I answer from folks as I'm giving away seeds or other things. Oh, yes, beautiful. Thank you. Okay, I'll go into a few quick resources, and then I'll deal with a bunch of the questions I saw in the chat there. These are a few books that I really recommend if you want to learn more. As I mentioned, this is easily a semester's course. Seed to Seed is a really good book for seed saving. I think it's the second edition that's come out. I believe we have a copy here. I'm pretty sure Big Island has one. If not, I think the library system does. Don't all go and rush on it. But there's copies online. Breed Your Own Vegetable Varieties. This is one of the books that made me into a plant breeder. It's beautifully written. She has a PhD from Harvard in biology. Gave up running a molecular lab to be a garden writer. And I think she does a really great job of making plant breeding accessible to the general public. It talks about a lot of the concepts I brought up, but in more detail and in really approachable language. You do want the second edition of this. The first edition doesn't have as much information. And she's written several other good books on gardening that are worth checking out. In terms of like the text that we would use for a semester-long course, the classic is Hartman and Kester's Plant Propagation, Principles and Practices. I did not check what edition is the most recent. I think this image says 9th edition. But if you want one that goes into way more detail, that's one to check out. And then if you're interested in tissue culture, this is the classic book on how to do tissue culture, Plants from Test Tubes. There's multiple editions of this as well, dating back from the 70s. This is what I learned from when I was doing my undergrad. I should, in all conflict of interest things, say that my PhD advisor, Mark Bridgen, wrote the most recent version and updated it because I think it hadn't been updated since the 90s. But this goes into lots and lots of detail about technique. It has a whole buying list of, if you want to start a lab, get this. And it has some specifics at the end about how to do tissue culture of roses, begonias, etc. Might not be something that, you know, you're going to be leaping up to do. But if you are interested, this is one worth checking out of, say, a library. And then maybe playing around with something like a kitchen tissue culture kit. Links to check out. I'm not going to spend too much time on these because we'll send these out to you. I mentioned the environmental impact quotient. Native plants, including the canoe plants. These are different ones to check out. There used to be other databases. Unfortunately, one of the ones I checked, the link was broken, but there's a bunch of propagation stuff online. Sift through it, you know, use some of the stuff I talked about in this lecture to kind of evaluate it and some of these terms. Also, I really recommend the CTAR publication database. I know I brought it up a few times today. It is the most locally applicable information, I feel like. And I bring up YouTube. I know YouTube is full of so much trash, but it also has some really good stuff there as well. Just kind of watch things with a grain of salt. And for things like grafting, ideally, you'll see an in-person demo. But if you can't, this is a really good way to kind of watch the same thing over and over. And that might be hard, you know, to ask someone in an in-person demo to do the same thing 15 times. But with a mouse click, you can watch them do it 15 times and really kind of get that detail from it. I mentioned that we'll be doing an advanced training on seed saving probably in April. This is a talk that Glenn Tevis, who's what they call an omni agent, does everything on Molokai, put together. I think he did it a few years ago, but he's updated it. It's now a 3-part thing on YouTube, on the Molokai YouTube channel, linked below. I think if you type local seeds for local needs, you'll also see it. It's playing, I believe, on some of the local TV stations as well. I know here on Kauai, I helped get it on was it Koiki? Something like that. I'm trying to remember. But you can also check it out on YouTube. I think Glenn will be involved in the one in April, and this talks about some of the local challenges with seeds as well. And finally, just something I came across in one of the plant groups online. You know, I think this is always worth keeping in mind, you know, as we kind of define ourselves differently. All right, I've been talking a lot. I'm going to sip on a little bit of tea and try to go back to some of those questions I saw from before.
And just keep in mind, we've got about an hour for a hands-on presentation ourselves, so we'll have to hop off at exactly 12. So that gives us about 5 minutes. Yeah.
All right, let me run real quick up then. I mean, yeah, I see a really good point.
Oh,
go ahead.
Just we can leave it from and go do Hilo things ourselves if you want to keep talking past 12 though, James. I'm just letting you know we're gonna hop off. Yeah.
Okay. Yeah, I'll try to keep it quick. But yeah, I see a few things. Yeah, you cannot buy GMO seed without some sort of certification and education about it. So chances are that you buy GMO seed is very, very slim. Some plants are sterile. That's, you know, as I mentioned in the beginning, some things are easier to propagate through asexual. Inbreeding and outcrossing, I'm going to say look into this a little bit more just because the explanation would take me a while, and look back on these slides. Inbreeding, basically the plant is okay pollinating itself. You won't have a problem. Outcrossing, it prefers to cross with other plants, even if they look differently, and will be happier in the offspring. Let's see. Was the wheat problem in existence prior to agribusiness? It was, yeah, it was a huge problem. It's still a huge problem in countries without seed treatment. There was a case in the Czech Republic of people losing 30% of their yields just a few years ago. You can vacuum pack seeds, you can. They do respire. I'm not sure of the longevity of them, but I would think in cooler conditions should be okay. Dehydrator, just check the temperature. You don't want to cook the seeds. Citrus clones be removed from seedlings. Depends on what your goal is in terms of whether you want something that comes true from the parent or not. And let's look down here at the bottom. Beautiful native plant propagation database. Glenn Tevis is amazing. He's going to be doing a few more talks. And the link to Glenn's thing. Yes, I will copy that right now, put it into the thing. And will also be in the slides when we send them out. All right, I can't see the time here. Yeah, still have one, maybe more minute. Any last pressing question on general propagation stuff? You also have my contact information. Like I said, I'm not going to go into detail for any specific plants just because we don't have time.
We get a question from Hilo. Let's hear it.
So,
Please.
you cut a tree like a coffee tree and you pop it because you want to have new growth.
Okay, so if you're cutting a tree like a coffee tree, like kind of coppicing it to get new growth.
What would you do to make sure it grows good without, because sometimes there's slower growth and faster. Do you cut the whole tree? Do you leave a branch so that it can get more uptake of fertilizer or whatever?
Ooh, that's going to be a hard one because so how this is more of a pruning question too. This is
Yeah.
A propagation, so maybe we'll answer that a little bit in a second section, and then when it comes to plants like that, you really do need to focus on the specific plant. Some plants you coppice completely and you don't have to worry about it because they're so vigorous, and then other plants like coffee, there's very specific protocols for pruning coffee so that you're leaving a certain number of trunks to allow for production of the next year and subsequent years, while pruning some off to make sure that there's enough airflow and you don't have non-producing trunks. So the answer is it depends on the plant.
Yeah. So, and I'll say I have a link, unfortunately not in front of me, on a pruning video that goes into some detail. I'll try to remember to send that out and put that up on the CMS site. Before Russell ends the meeting, I'm putting the Zoom link for the Kauai training. It's not going to be a long training today. We'll integrate into some more, but I'll talk about that. But if you all need it for the Kauai folks, otherwise I think we're all going to go off to our own islands and do our stuff.
Thank you.
Okay, thanks everybody. Thanks, James. That was a long talk. So go drink like a gallon of water there and don't talk.
I'm double fisting tea right now, so thank you.
Awesome, James. That was an incredible lecture. Really well done.
Thank y'all. It's fun to give. I love sharing with y'all. So,
And then welcome class. Let's join on pretty soon, okay? Just a quick stretch and let's join on.
Yeah, Kauai, same thing. Maybe 5-minute break just so I can drink some more, and then I'll see y'all. Okay.
This is, yeah, okay, little break, folks.
Awesome. Bye, everybody. Thank you.
Yeah, what's up?